Welcome to the Foundation of Neuroscience lectures. I'm Terry Signowski. So you may have noticed that there has been a couple of revolutions over the last few years. And I'm not talking about revolution somewhere in Ukraine or no, I'm talking about revolutions in science. What's really hot right now? Okay, put up your hand. You know the answer.
How do we define the revolution?
Revolution means that everything is different when it was like, it was maybe 10 years, 10 years or so. 10 years. Okay, we have some there. Okay, no, yes, go ahead.
Deep learning and neuroscience become mainstream. Many subfields.
Well, there you go. Actually, that's two revolutions.
That's true. Sorry, I know like the.
No, I was going to make that point that in fact,
Deep learning is basically there are many subfields of neuroscience. Like very standard.
yeah, well, deep learning is being used by the world, right? It's not just neuroscience, but neuroscience is having its own revolution, and this has been going on for about 10 years since the Obama's Brain Initiative was announced. And I was at the White House actually in Easter when he announced it. It was very, for me, moving to see the President of the United States get in front of the country and say that his grand challenge in science and engineering was my life's work. Now, nobody knew how it would play out, okay? And I'll just give you a very brief, let's see, is Grace here? We have two visitors from NIH who will be here until on Friday, but in any case, I was on the president's, this is the, not the president's, the advisory committee to the director of NIH, Francis Collins at the time, to give advice about the Brain Initiative. Obama had said that we're going to do it, and it's not just NIH, it's NSF, it's DARPA, FDA, a lot of the government agencies got involved, and the then head of NINDS, one of the the parts of NIH, which is primarily NIN neurological diseases and stroke. Okay, I don't know why they call it that because it's really neurology.
Exactly.
Or no, yeah. So
It has to do with who lobbies artists. A NIH institute is established by Congress. So the difference between centers and institutes at the NIH is whether the NIH director established it or whether Congress established it. And the names of the institutes go with the establishment.
okay, this is what happens to bureaucracy when you have rules within rules within rules. But
It doesn't matter.
So Story Landis was the director of NINDS. She told me, she's an old friend of mine. When I was a postdoc at Harvard Medical School, she was an assistant professor. She rose to the ranks. She said she had been trying to lobby for neuroscience for a decade. And it was at the low end of the totem pole, right? What's at the top? Cancer, heart disease, right? I mean, all of these really big killers, right? And she said, she gets a call from Francis Collins saying, the president has given me orders that I'm supposed to be the director now, leader here of the Brain Initiative. I don't know anything about the brain. Can you come up and give me a lesson? Then, a couple weeks later, he shows up on Colbert. You may know who Colbert is? Colbert Report. Okay. And he is wearing an EEG cap. But some obviously made EEG recordings on the background. He did a great job of explaining to the public what this is all about. He did a great job. It was
That's
really,
his job.
what?
That's his job.
Oh, okay. Then he's doing his job well. Okay, so why was it? It was widely successful. We went from recording one neuron at a time, and maybe a few more if you had a little array. But now we can routinely record from tens of thousands, 100,000. The record is now 1 million neurons simultaneously. And this is from dozens of parts of the brain. And that has revealed things that nobody had ever imagined before. And, you know, I'm going to talk tomorrow morning about traveling waves. That's something that, you know, you can't see with one electrode. So in any case, these two areas are converging. And, you know, as AI gets more and more and larger network models, it can do more and more amazing things. And we're learning things, as you'll see, later in the week, that we can learn things from the people in AI about how they attack the problem of trying to understand how large language models actually work. And at the same time, as neuroscientists make progress in understanding how the brain actually solves those same problems, maybe they'll be able to help each other. And it's not going to be everybody. Obviously, there are a lot of engineers who are basically in the trenches trying to get the thing to work more efficiently and so forth. And there are a lot of neuroscientists who are just in the trenches trying to understand what the signals mean and analyzing the signals. I mean, those are all good things that are being done, but there's a smaller group, and some of them are here, who are trying to mesh the two. And the neuromorphic engineering is actually, as you know, it goes back to 1989 roughly. And that was the goal. That was the goal, the initial goal. So they were carving this way ahead of everybody else, right? And now his time has come. Okay, so let's introduce a few of the people that are here. I will introduce Michael in a moment, but let's see. Barry, where are you? You should introduce yourself.
Yeah, I'm Barry Richmond. I work at the intramural program of the National Institute of Health. And
NIH?
That's
By the way, that's mental, NIH is mental health, and it used to be called psychiatry.
But the goal of my research has been to take behavior at the systems level and understand how it's being generated by the circuits in the brain. And we've been using a lot of the primitive versions of the modern tools for a long time.
Okay, good. And let's see, Andy.
Andy, I'm a fellow at the Harvard Cancer Institute. Trying to use
What is that? I've read about it, but I have no idea what it is, who's funding it, and why are you there? That's the
yeah, funded by Mark Zuckerberg. He donated money. And the mission is understand natural and artificial intelligence. So I'm trying to be at the intersection.
Okay, how big is it? How many people?
Trying to hire something like 10 or 15 faculty. We have 8 fellows each year, so there's another 8, 24 fellows.
So they're trying to build it up basically. Okay, so it's still in its infancy. In fact, you were probably one of the first fellows we built.
Yeah.
Okay, and then what happened to you in your life before that?
I did my PhD in Amsterdam, University of Amsterdam, with swelling on generative models and machine learning side, but then kind of taking the direction of computational neuroscience, seeing how we can add components like topographic organization or interesting neural dynamics such as traveling waves or oscillations to these models, and then use traditional deep learning techniques to see if we can optimize them to do interesting things.
Okay, well, you'll be hearing from Andy tomorrow. We're having this week 9 a.m. lectures. Michael Berry is the first. I think I'm talking tomorrow morning, and then Andy is talking tomorrow evening, right? And let's see. I think you're talking tomorrow night. When? Tonight. Oh, okay. So you'll be hearing from Barry about working memory. Working memory is one of the themes. I'm going to talk about working memory too. Okay. So it's really exciting right now because we actually have some theories for how working memory actually works. Okay, I guess anyone else from our group here? Not yet. Steve Zucker is coming on Tuesday. Yep. Oh, John, why did you put your hand up?
I thought you'd remember.
No, I of course I remember you. I didn't see you.
So I've been at Caltech for like 40 years. I have my own little department called Control Data Systems. I'm a mathematician, and I have a very, very loose relationship with reality. But that won't, so if you're talking about any topic, I will have very strong opinions about how things should work. And then they may or may not map onto how they actually do work. So.
But John always has good ideas, regardless of whether or not they work.
Well, the thing is, yeah, a good idea doesn't have to be right. It has to put you in the right place in the frontier. So anyway, I'm good at that, but I'm not good at being right, so don't trust what I say.
Okay, so so in any case, it should be a fun week and we'll be around. So Michael Berry is at Princeton and he's in the neurobiology, actually Neurosciences Institute. And then is it a biology department there?
No, it's just the institute now. It's kind of.
Oh, really? Okay.
Sort of.
So I did my graduate at Princeton in physics. And there was no neurosciences institute. There were a few scattered neuroscientists in biology and psychology and engineering, actually. And it's all changed radically. And I can vouch for that because I was there at the end of May to receive an honorary doctorate from Princeton. It was a great honor. And I could not recognize the campus. This neuroscience building is huge. It's like this monstrous thing that's growing from the ground. I mean, it wasn't there before, right? You know what it's like if you go back to your childhood home and you see it's changed. It's kind of jarring. But this is the future, and so Michael worked for many years at the bottom of the visual hierarchy in the retina. You can't get any lower.
Or higher.
Or fundamental.
But as he was pointing out last night at dinner, he said, people are talking about traveling waves and dynamics and everything. Well, it's already happening at the retina, right? You don't have to go to the cortex. And he actually made a lot of discoveries along those lines when he was focusing on that. But now he's gone up in the world, and he's now thinking a little bit more about how the cortex and the thalamus, as you'll hear, are gathering the information coming in from the sensory systems and also integrating that information. And it's a very difficult problem. We need new ideas. Michael.
Okay, thank you, Jerry. Okay, so yeah, just following on that thread, so after I decided to kind of move on from the retina, start thinking about central brain. One of the first things that hits you is essentially everything is connected to everything. The retina was sort of nice to study because it's this isolated outpost of visual processing. In the central brain, there are loops and feedback, so it's really not very conceptually easy to separate out, different regions of the brain and kind of think of them in isolation. So I've been, trying to develop this research program I call large scale brain theories, that attempts to broach, what are these kind of interacting regions of the brain doing? Okay, and so what I'm going to talk about right now is the loop between the thalamus and cortex and some of the functions that it might be carrying out. Okay, and so like the underlying approach for this work I'm doing is kind of this idea of repeated microcircuits. Like especially for neuromorphs, the brain has just this bewildering or kind of alphabet soup of different brain areas, and these brain areas all kind of apparently do different functions, but in many cases, if you look at kind of the underlying cell types and how they're organized into layers and circuits, you know, there are many, many regions of the brain that are nearly the same, you know, like for instance, the neocortex. And so, you know, so kind of a simplifying assumption is, you know, to kind of look at the microcircuit of kind of this collection of different brain areas. You know, try to understand you know, it's kind of anatomy and biophysics in a lot of detail, and then you know, kind of make this assumption that it's primarily kind of repeated many places in the brain. So you know, that allows you, that assumption, you know, allows you to kind of make a lot more progress than you could if you needed to have a detailed story about kind of every different brain area. Okay, so there definitely are, you know, biological differences that exist between, let's say, different areas of the neocortex. But you know, the basic idea is, you know, perhaps this is a useful approximation at the moment to try to understand kind of more global brain dynamics. Okay, so and again, I think especially for neuromorphs, you know, one kind of important, I think, simplifying fact is this idea that, you know, all the different brain areas can be organized into 7 major brain divisions. You know, so and then, you know, here they are. And for 6 out of the 7 major brain divisions, you know, except for the brainstem, they have repeated microcircuits. Okay, so at a certain level of approximation, you know, maybe what we need are kind of like, you know, 6 or 7 different stories about, you know, kind of function of very different areas of the brain, and you know, maybe that'll take us quite far.
So it's interesting, brainstem is one of the most ancient.
Yeah.
Which it got us off the ground, right? Early vertebrates. So maybe that's why it's not as well organized.
Yeah, I mean, yeah, I mean, you know, there are a bunch of kind of very specialized kind of sensory motor systems in the brainstem. You know, they do kind of specific useful tasks. You know, they're not that plastic, not that flexible. You know, so kind of, and they evolved earlier in evolution. You know, so I guess part of the story here is like maybe these other sort of higher brain areas evolved a little bit later in evolution, and then, you know, one thing that's easy for evolution to do is to kind of increase the size of a brain area. And so just proliferated these brain areas kind of virtually the same microcircuit. And, you know, these particular microcircuits are just doing very useful things. So, you know, it all was very useful. Yep.
The microcircuits for each one of these areas are not going to be necessarily the same.
No, they're absolutely different. So that's a very important point to make, is that, you know, the cell types, the circuits, the computations among the major brain divisions are absolutely different. You know, so in that light, you know, I think we're probably never going to have like a unified theory of brain function. You know, we're going to probably always need to talk about like maybe 7 different things. But that might do a good job. Yeah.
Is it okay to think of this, you know, in terms of trade-offs, as fast and old at the bottom? Because we basically was saying spinal cord is a problem. And then as you go to the top, you get more accurate, more predictive, but a lot slower.
Yeah, on a certain level, yeah.
Spinal cord has a lot of flexibility, actually.
It does. But I mean, in general, these brainstem circuits have less flexibility.
Well, they control things like sleep, right?
Yeah, that's one.
And you know, you get things that like hypothalamus and you know, which basically are responsible for, it's like the operating system of the brain, right? Vegetative functions. That's really demeaning. Something that's really important.
Yeah, but you don't have any.
By the way, this is what large language models need. They need a brainstem.
Yeah, exactly. This is, you know, so you know, when you think about the more abstract function of these you know, higher brain divisions with repeated microcircuits, you know, the issue is like they never have to learn the really nitty gritty of, you know, was this thing in my mouth like nutritive, yes or no? Like the brainstem, you know, figures that out and gives that information to the higher centers. It doesn't have to learn that. Yeah.
This is obviously the mammalian picture. In birds, for example, it's kind of one big brainstem.
No, I mean, these divisions are all there in lower vertebrates except for neocortex.
But let's say birds don't have layered area section.
Well, what about techno?
Well, they, yeah, I mean, so most of these brain divisions don't have like multiple layers. It's kind of the neocortex with multiple layers. So yeah, so this is for mammal, but for lower vertebrates, you replace neocortex with like the pallium, which is archicortex. So it's actually, you know, pretty similar.
But by the way, Harvey Carter, who's
Yeah.
at UCSD and is, you know, one of the world's leading experts on bird brains, realized that if you look at the connectivity, so instead of having the layered, you know, cortex, it has a bunch of discrete nuclei. But if you look at how they're connected, they're actually connected in this way that is similar to the different layers in the cortex. And if you actually record from them, discover that the kind of information that they have in their processing, it's just another way that the evolution organized the very same algorithms in a way that you can't see just morphologically. You really have to look at the circuits in order to be able to tell. And by the way, it turns out that what convinced everybody, now all the bird people now agree with it, by the way, right? When he started out, they thought it was crazy. But now, the way that they were convinced is by, if you look at the development of the brain, there's a whole series of stages that neurons that you know the different areas have to develop, and new neurons are being created. And it turns out that the very same precursors of the neurons for the neocortex are the ones that are precursors for these different nuclei in the bird. So in a sense, it was nature had this old plan, going way back, you know, to the frog, that was basically elaborated on, but it elaborated in different ways, but same algorithms. And this is actually an important lesson, which is that that morphology, although it's a good place to start, doesn't give you the truth. That you really need to understand not just the how they're connected, that's a circuit, but also the information that is being represented and the algorithms. It's the algorithms that are going to give the game away. Oh, by the way, I forgot to introduce both of you. I just realized we have two more people here.
Sorry, Michael.
Sorry, Michael. This is an ectopic intro. Okay. Okay,
No
Mackenzie.
worries. Yeah, hi, sorry, Mackenzie Mathis. I'm at EPFL in Switzerland. I work at the intersection of neuroscience and AI, specifically working on adaptive intelligence, trying to understand how we can rapidly change our behavior in light of changes in the environment and circuits. So we work on core machine learning algorithms, computer vision, and I do a bit of systems neuroscience. So I'll talk Wednesday morning. Thank you.
Okay.
I'm Alexander Mathis, assistant professor also at EPFL, and I'm interested in understanding the structure of behavior, and then also the algorithms that give rise to behavior.
So, Bill Newsom, when we were writing our report, by the way, this is for brain issue, came up with a great kind of the soundbite, right? We called them in journalism. Nothing in neuroscience makes any sense whatsoever except in the light of behavior.
Yes.
And you ignore behavior at your peril. That's the problem. If you just kind of get lost in the circuit, you know, you'll never figure out why it's there.
Change the way the scene.
You're in good company.
I don't think people believe that. Yeah, so just briefly picking up the thread about birds, you know, so this work kind of it's just kind of being more seamless about the addition to my talk. You know, the interesting story that kind of emerges from this anatomical work is basically so the idea is that mammalian neocortex kind of packages a set of circuits together all within the same repeated microcircuit in a way that you know, all you need to do in evolution is like grow it bigger, and it can kind of organize in a very useful way. But for the birds, since these different microcircuits are scattered in different nuclei, you would essentially need kind of many mutations lining up to produce like a new kind of hierarchical level in the bird's brain. So the mammalian neocortex is very highly scalable, although it, so in a sense, it probably embodies many of the same functions as in the palate.
So
Okay, but
birds are the last living group of, going back to the dinosaurs, right?
Well, yeah, I mean, birds are, yeah, I mean, dinosaur brains they're probably a mix between mammals and birds. It's a little, they're, well, why don't I move on? Okay, so in the light of, thinking about these 7 major brain divisions, one of the things I've done is I've tried to come up with like 1 or 2 catchphrases to describe the computation that occurs in these different brain divisions. So we'll start with neocortex. So the 2 catchphrases: engine of unsupervised learning. That's kind of a sensory processing story. And then the other one is generator of motor plans. And that's what I'm going to focus on for this talk today. Okay, so where are the motor systems in the neocortex? They kind of proximally arise with this super important cell type. It's a pyramidal cell in layer 5, has kind of multiple names, layer 5B, because it's at the bottom half of layer 5. Layer 5 PT for pyramidal tract, because its axon descends to the brainstem and spinal cord. Or layer 5 tufted, because it has a gigantic apical tuft in layer 1. Yeah.
Sorry, can I ask a very foolish question?
Yeah.
What does layer 5 mean?
Well, neocortex can be divided into 5 layers. If you look at the, kind of the size of the cells. This goes back to like Cajal and early neuroanatomists. Just so one thing in particular, like this cell type has the biggest soma of all the pyramidal cells in the neocortex, and I'll show you a really beautiful picture of one of these cells.
But you have an earlier slide. Why don't you put it back up with the layers?
The first slide.
The first slide. The
Yeah,
first slide.
okay.
Yeah, because that's an important
Right,
question.
there you go.
There
Yeah,
it is.
so
those are the layers.
Yeah, so these are different layers. This is kind of one level detail of the neocortical microcircuit, and I didn't want to go into it in too much detail because I'm really only picking out layer 5 for today's talk. Okay, so the basic issue here, I guess star means this is an interpretation that I'm making, but others have made it. If these axons go to brainstem and spinal cord, like they're encoding a motor command. Okay, some evidence for that. So just to briefly mention a few things, some evidence comes from microstimulation of primary visual cortex. If you do that in the superficial layers, you get kind of disrupted visual perception and kind of phosphenes. If you do the same stimulation in deep layers, you actually get eye movements. Okay. And then another brief evidence, Tony Zader has a bunch of nice papers studying, this cell type in primary auditory cortex and looking at its projection striatum. Yeah.
Just when you talk about what it generates eye movement, is it generating eye movements through motor plan or because it has a sensory consequence that's so compelling you make an eye movement?
Well, I mean, I think this difference argues that it's directly stimulating the motor pathway. Because look here, you're stimulating the more sensory regions, causing effects to sensory perception, and you get no eye.
So another dumb question.
Yeah.
What are phosphenes? Are they chemicals that are created?
No, they are also chemicals, but so what a phosphene is it's kind of this ghostly nebulous visual percept. So like the way you can see that for yourself is if you like press on your eye, like close your eyes for a little while, press on your eyes, and the pressure fires retinal ganglion cells in a random pattern, and then V1, the rest of your brain tries to do something with that, and it's a mess, so it produces this kind of phosphine. So that's kind of a general consequence of activating a group of neurons in, let's say, a visual pathway that like don't really belong together.
So I think, people are trying to, with visual prosthetics, to actually use those electrodes in the visual cork V1 to try to help people see, right?
Well, that actually works very badly. Because you get these, because basically the neural populations in V1 are not organized well to stimulate with a single electrode. So it works much better in the retina. Okay, question? Yeah.
And I have another simple question. You said the L5D
Yeah.
is one of the largest cells.
It's
Is that
yeah,
because
it's the largest. Yeah.
It has to drive axons very long distance to the spinal cord.
Yeah, exactly. So
it really matters how far you send.
exactly. That's a general principle you see all over the brain, that the neurons that have the biggest soma have like the biggest axonal arbor. And like the basic reason for that is the axonal arbor needs all these chemicals from the soma to keep it alive and functioning. And so you need more of those chemicals. So you need a bigger soma. Okay, so back here to primary auditory cortex. So so they studied this simple task where a rat associates an auditory cue, high frequency or low frequency, with either licking left or licking right to get a water reward. And so what they showed is if you use channel rhodopsin to stimulate these layer 5 neurons, you bias the animal's choice. Okay, so it's showing that those neurons have a causal role in actually producing the licking
channel rhodopsin.
What?
You typically not know what CHR2 means.
Oh, yeah, there's channel rhodopsin 2, so who knows what that is? Okay, more than half. It's some people have called it God's gift to modern neuroscience. It's a protein found in blue-green algae. You put it on the cell membrane, and if you shine blue light on it, it opens an ion channel that causes a depolarizing current. So you can cause action potentials in the neurons that express this protein. And because it's a protein, you can use all the tools of mouse genetics to express it in specifically certain cells that you're interested in, and then stimulate them and see what the effect is. And so experiments like that show that this population of neurons are actually playing a role in driving the behavior in this particular auditory task. Okay, so
Is it not really biasing the choice, because those neurons will go from left, right, but also those other things.
Yeah, so what's going on here is the in this looking left, looking right, the animal's brain is kind of it's bilaterally organized. So basically the A1 and striatum in the left hemisphere will trigger a rightward lick, and so if they stimulate there, they get more rightward licks. It's the kind of the contralateral organization of this particular movement in this case. It's not always true, but it's true in a lot of situations.
I'm sorry, I know you want to get to next
Yeah.
slide, but I'm just gonna challenge the notion that this encodes motor commands. That like this isn't
Yeah.
evidence to me of motor commands. This is evidence that you can change sensory action circuits to drive behavioral changes. But like V1 doesn't project to the spinal cord.
V1 projects
Superior colliculus.
almost entirely to superior colliculus,
Exactly. Yeah.
which makes movements.
It does, but it does other things too, right? It also has this like direction
Yeah.
activity. It's involved in reaching in primates. I don't know if the Schiller work would say that's a motor command V1. This would be the first time I've ever heard of a neuroscientist claiming that this is a motor command.
Oh, well, I mean, it's not original to me. It's there in the literature.
Well, I mean, but it's a loop, and
Yeah.
where along that loop does it go from a sensory trigger to a
Yeah.
motor command? It must be
Yeah.
somewhere along that pathway.
Yes, that I agree with, but evidence that this is driving muscle level motor commands, I don't.
No, it's not driving the muscles directly.
Exactly. So
Okay,
maybe define
so-
motor commands is the question.
Yeah, so in the context of trying to tell the story in a limited amount of time, why don't we discuss this afterwards? And so I mentioned this is an interpretation, but I think it's a very good one, lots of evidence. But we can debate that more. Also in these experiments, during learning of the task, there was kind of potentiation of the cortical striatal synapse in a way that, completely made sense with the animals, changes in their behavior. Okay. Okay, right. So very important fact about the cell population is it's present in all cortical areas, sensory cortical areas as well as motor cortical areas. And so I think of this as maybe the most important fact about the brain that many neuroscientists don't seem to take into account. I think neuroscientists these days know this, but I think a lot of the time it just doesn't factor into how they think about how the brain works. I think it's super important, in part because you immediately get a serious problem. If every neocortical area is kind of cooking up its own motor plans, in principle, you could trigger multiple incompatible movements, and that's really bad, like could lead to death quickly, so there must be a way of preventing that. So I'm going to argue a solution is kind of thalamus and loops with the cortex. Okay, and so I also want to mention that these same cells also project to striatum and cerebellum to form this kind of massive kind of network in neocortex for motor planning. And all of these brain areas, or actually striatum and cerebellum, both feed back to different areas of the thalamus that feed back to the cortex. So all these systems form like kind of massive, powerful recurrent excitatory loops. Okay, I just want to throw in a picture of one of these neurons. Very beautiful fill made by Japanese neuroscientists. So here's the LAIR5 cell in cortex. Goes down in striatum, motor thalamus, superior colliculus, bunch of basal ganglia, pontine nuclei, cerebellum, blah blah blah. That's like one neuron. Okay. Okay. So now let's move on to thalamus. So the catchphrase is communication hub of the brain. So these two little red football-shaped objects are the thalami.
They're under the cortex.
Yeah, they're under the cortex. It's a bilateral, so you have one thalamus for each hemisphere of the cortex. Okay, so thalamus, I think at the moment is really one of the most kind of mysterious parts of the brain. There are a lot of different ideas about how to think of the thalamus. What you primarily find in the literature is like a big alphabet soup. So there are all these, ventrolateral, ventral anterior, lateral dorsal nuclei. But I'm going to argue we can really think about it as falling into kind of 3 or 4 major divisions of thalamus. Core, matrix are the 2 main. Intralaminar, I'm going to mention, but I'll talk about. Reticular nucleus are kind of a shell of inhibitory neurons on the surface of the thalamus. Okay, so there are kind of there have been 2 main approaches to kind of understanding different thalamic nuclei. From Murray Sherman comes the idea of first order nuclei versus higher order thalamic nuclei. So the first order thalamic nucleus gets subcortical input as its driving source. So for instance, the LGN gets input from the retina. The MGN gets input from the auditory brainstem. Okay, so those are kind of sensory gateways to the brain. The other type are higher order nuclei, and for these thalamic nuclei, the driving input comes from the cortex itself, from these LAIR5B pyramidal cells. So examples: ventrolateral, which is a major part of motor thalamus; medial dorsal connects to the prefrontal cortex. Okay, so it turns out almost all of thalamus is higher order. Okay, LGN is the nucleus we know by far the most about, but it's really not representative of most of the rest of the thalamus, and so I'm going to focus on kind of higher order Okay, another important distinction comes from Ted Jones. So he kind of used some kind of molecular labels to find some different cell types in the thalamus. And a very important difference between these cell types has to do with where they project in the neocortex. Okay, and let me see, I'm gonna, yeah, sort of a more recent articulated version of this comes from Fernando Klaska, where he talks about 4 major types of thalamic relay cells. Okay, so the core type, you know, is a relay cell that projects kind of very strongly into layer 4, the input layer of neocortex. It has some other minor collaterals. And focally means it has a kind of a tight spatial region. So this is what you see mostly in the LGN. Okay, so roughly speaking, this is a sensory relay. Okay, but you have a couple of these types of matrix cells. And what's prominent about them is they have huge input into layer 1. And this pink one, the multi-areal, also has a big input into layer 5. You know, this is actually the major cell type in the motor thalamus. So I'm going to mostly be talking about that one. And then there's the intralaminar cell type that has inputs into kind of layers 5 and 6. So I'm going to mostly ignore that in this talk. Yep.
So like the cortex, it seems like thalamus is more, there's no organization, lateral organization or layer organization.
No.
It's just a bunch of blocks.
It's just a clump of cells, you know, like the nucleus. It has this reticular nucleus, you know, over it, which has a whole bunch of inhibitory neurons. It has a few local inhibitory neurons, but in some nuclei it has like actually none, you know, so I'll talk about this in another slide, but the thalamus itself as a local circuit is incredibly simple. It's the simplest part of the brain by far.
So I think it's worth pointing out that on the right, you have a nice little diagram with colors.
Yeah.
And each of those is a different thalamic nucleus that projects to different parts of the cortex, right?
That's right. So yeah, so I mean the origin of the alphabet soup is that there are there's kind of modular connectivity between thalamus and cortex. So one cortical area will send its projections to kind of one nucleus of the thalamus, and that thalamus will send almost all of its projections back to the same area of cortex. So you have a bunch of these kind of repeated sets of Thalamo-Cortical loops. Okay, so yeah, so that's, yeah, I mean, so this alphabet soup is describing, you know, that type of detail. So for instance, VL and VM, ventrolateral, ventral medial, are the 2 main parts of what are called the motor thalamus. So they have reciprocal connections with the primary motor cortex. And you know, here's medial dorsal that I talked about that connects to the prefrontal cortex. Okay, so there's all this detail, but you know what I'm arguing is, like, if the neocortical microcircuits are like kind of the same repeated microcircuit, then all of this detail is maybe not that important. And what's more important are these couple different cell types, and that's important because of where they project. You know, this cell type projects to the kind of the input layer of neocortex, whereas this one projects to kind of layer 5 and layer 1. Okay, so yeah, so LGN is you know, by far the best understood nucleus of the thalamus, and so Murray Sherman and others have made this important distinction between driver inputs and modulator inputs. So the interesting thing here is, here's a relay cell in LGN. If you just count all the synapses onto it, 5% of its input synapses come from the retina, and about 50% are descending inputs from the neocortex. Okay, and the remainder are ascending inputs from the brainstem that are neuromodulatory. Okay, so But when you look at the like receptive fields, it turns out these neurons have like the same receptive fields as retina. You know, it's a very, very good approximation. So basically what's happening is this small subset of synapses are really dominating the firing of this LGN relay cell. And so, you know, Murray Sherman has called them driver synapses and these other ones modulator. Okay, so synapse is very different. So it turns out the properties of the synapses cluster really extremely nicely. So here's kind of these two clusters, drivers and modulators. So drivers have a bigger first EPSP, more depression, and no metabotropic glutamate receptors, whereas the modulators are kind of the opposite for all those. So the point is, you know, there are these two classes of synapses, and the drivers are mostly determining the firing of the LGN relay cells. Okay, here's just more anatomy. These are inputs to thalamus from layer 5 of neocortex, and these are the layer 6, so drivers, modulators. Okay, so right, so the idea is the driver inputs, they're strong, they fire spikes in the relay cells quite deterministically. It's not clear if there's any plasticity in those synapses. You know, retina and LGN definitely no. Maybe it's an open question in other parts of thalamus, but maybe not. Modulators, you know, weak influence, they can kind of somewhat boost the firing of cells that are going to fire anyhow. They have a lot of synaptic plasticity. Okay, so I'm kind of mostly talking about the driver inputs in this story. Okay, so what is the computation in the thalamus? It's basically lateral inhibition. Okay, so it's really quite notable. These relay cells have no excitatory synapses onto each other. There's no recurrent excitation. There's almost no kind of local circuitry. So you get input from the cortex and you pass it back to the cortex. Okay, but there is lateral inhibition. So some areas have local inhibitory interneurons, but the main thing is the nucleus reticularis. Okay, because all of the thalamic relay cells send collaterals to the nucleus reticularis, and then that feeds inhibition back onto the thalamus. So, you know, different excitatory thalamic relay cells are always kind of mutually inhibiting each other through the nucleus reticularis. So lateral inhibition. Yeah, so LGN, you know, roughly half of the retinal spikes actually cause an LGN spike, it's lateral inhibition. Okay, and then the other major computation is the feedback to the cortex. So kind of the main thing thalamus is really doing is through its feedback to the cortex. Okay, so let's think about that feedback. And we'll consider these multi-areal relay cells. Their output goes to layer 1 and layer 5, forms these recurrent excitatory loops, of course. Okay, so let's think of assumption here. Let's think about the plasticity you're going to get in this pathway. Okay, so the idea is, you know, let's imagine there's a subset of these layer 5B cells that encodes one particular movement. Okay, so I call that layer 5B star. It's, you know, that's population code for one movement. So, you know, those spikes go into the thalamus and they deterministically fire a subset of relay cells in the thalamus. So I'll call that thal star. So those are the relay cells associated with one movement. Yeah.
Just a quick thing on a previous question. When you say encodes something motor, you don't mean individual muscles, you mean complex.
Yeah.
Very complex, very high layer, must multiply be coordinated by a lot of lower.
It doesn't have to be that complex. It could be lick left, lick right.
It could be.
It could also be complex, yeah.
But it could be like turn right, you know, on a path.
Yeah.
Yeah, okay, I just want to.
Yeah.
Because you're using movement, I like it, but you're using movement very broadly.
Yeah.
And some people might think of movement as just being my finger. You're thinking about being the whole body.
Yeah, I mean, so some of these axons go down to spinal cord in some regions of the neocortex where they directly synapse on spinal motor neurons, you know, so So in some cases, like, these cells can actually cause like a muscle twitch.
Yeah, but they don't, they could be quite complex.
But in general, they're causing like kind of a more complex movement.
I have a dumb question. So this is like storing, this is an architecture and a program is being stored somehow, right? In some way, as a computer science.
Yeah, I mean, so I guess what I'm gonna argue here, I haven't finished this argument, is that I'm gonna assume a certain type of plasticity has like already occurred, okay, and that's in some sense like the program maybe that you're thinking about, and so then like assuming that I'm gonna explore like a model of the dynamics. Okay, so okay, so kind of the key idea here is you have this defined subset of relay cells in thalamus, they feed back to cortex, and of course this subset of LAIR5B cells were just firing. So these two types of cells are firing together, and so you're going to potentiate those synapses. Okay, so the idea is in this feedback pathway, you're going to have extra strong excitatory synapses onto the very same LAIR5B cells that drove those thalamic neurons in the first place.
How quickly does this potentiation happen? How quickly does this potentiation?
A little hard to say, so
Is it one kind of seconds or?
it could potentially be seconds, yeah. But you've had like your whole lifetime up until now to be doing this. So the basic idea is, let's imagine that over time in the LAIR5B population, it's sculpted into certain clusters of subsets of cells firing together for one particular movement. Then if you do that movement, 100,000 times up until now, like you're going to be able to strengthen this pathway pretty specifically. So, if you're confronted with a new situation, I would say that this doesn't evolve very quickly. Yeah.
Should we think about the thalamus here as a dynamically reconfiguring the L5 cells to really perform certain things, or?
Okay, I'll get to a model of that.
Yes.
Okay, so the consequence is it forms a specific amplifier circuit. Okay. Yeah. Okay. And the idea is this combined loop, Thalamo-Cortical loop, or TC loop, I'm going to call it, represents a specific movement. Okay, so now if the brain is considering multiple movements at once, what basically happens is a winner-take-all dynamics through inhibition in the thalamus. Okay, and so let's say these are two populations of cortical LAIR5 cells encoding two different movements. They could have inhibitory circuitry between them in the cortex, but in general they're separated very far. When you bring the signals into the thalamus, they're like right next to each other, and so you're kind of guaranteed to have this lateral inhibition in the thalamus. So they compete, loop with the most activity wins. So each loop is trying to amplify itself and shut down the other loop, and so you get a winner-take-all dynamics. The winning loop then triggers its movement. I'll mention a little bit more about the threshold for that in a moment. And it suppresses the other loops. Okay. So the idea is that this gives rise to a system of kind of global motor coordination, where among all the different motor plans that neocortex is cooking up all over the place, they're all competing with each other in the thalamus, and only one of them can win. So that's why you don't generate multiple incompatible movements. Yeah.
So are you saying some loops get shut down?
Yeah.
By other loops?
Yeah.
Okay.
Exactly. I'll show you in a moment. Okay, so I'm going to present a computational model of this, but okay. Before I do that, I want to just mention a couple things. First of all, I want to show you some important experimental data about the importance of the thalamus. And then I'm gonna talk about what determines like when you win, and that has to do with the basal ganglia. Okay, so quickly, importance of the thalamus. So the Swoboda lab has done a series of really nice experiments like this. So they're focusing on this one simple task. So the mouse is sitting here in darkness, and what happens is there's kind of a machine that has a post kind of come up either nearer in the blue position or farther in the red position. So what the animal has to do is it has to use its whiskers to feel, is the post in this kind of closer or farther position? And then depending on that, it licks left or licks right, okay? So so kind of previous to this paper, the lab has done a whole bunch of experiments where they show that if you photo inhibit this one cortical area called ALM, you knock out this behavior quite strongly, okay? So it's cortical area ALM that's doing this behavior. And so in this paper, then they're looking at the corresponding area of thalamus. And so one thing they did is they photo inhibited the thalamus, okay? And that, you know, kind of strongly knocks out behavior, just like inhibiting the cortex. Okay, and just to mention here, again, this is a like a kind of a contralateral behavior. So left hemisphere drives a rightward lick. So if you inhibit there, you knock out the rightward lick, but you don't affect the leftward lick because it's the other hemisphere that is kind of driving it. Okay, so thalamus is required for the behavior, just like this cortical area is. If you look at the record from neurons in the thalamus, it's a little bit hard to see in this data, but they have a bunch of ramping behavior during the sample period. Then they have maintained activity during the delay period, and it's substantially similar to the activity that you see in cortical area ALM. Okay, so now I think the most interesting part is kind of looking at the dynamics of what's going on when you inhibit the thalamus. So what they're doing here, you know, photo inhibit the thalamus, record from neurons in ALM. And so time 0 is when you inhibit the thalamus, and activity in ALM drops to 0 with the time constant of like 10 milliseconds. Okay, so without this thalamic area, you just shut off the cortex. Just showing that photo inhibition works in the thalamus itself. Here's one other interesting piece of data. Now they're recording the membrane potential on some neurons in ALM. So if you inhibit the thalamus, the membrane potential kind of crashes very fast. If you inhibit M1 motor cortex, which has a huge set of connections to ALM, you get almost no effect. Okay, so for this area ALM, it's really inputs from the thalamus that are dramatically more important. Okay, so showing this data to help convince you thalamus is super important for cortical activity and for behavior. Okay, so now switching gears a little bit, I'm going to talk about this kind of threshold for deciding which loop wins. So that has to do with the basal ganglia. And basal ganglia is this funny shaped thing, at least in a human. In a mouse, it's just like a single lump. But it's a bilateral brain structure kind of, again, underneath the cortex. One thing it does is gating of voluntary movement, which I'll talk about. It also is the kind of the originating source of reward learning or motor learning, but I'm not going to talk about that here. Okay, so there's this this basic kind of Rube Goldberg apparatus where these layer 5B neurons, the very same neurons, branch and send that same motor command into the striatum, where they fire neurons there. The striatum has inhibitory neurons, which inhibit the pallidum, which is normally spontaneously active, which normally inhibits the brainstem. And so this input from the cortex fires striatum, pauses pallidum, and releases movement in the brainstem. So yeah, so the pallidum has neurons that spontaneously fire at a relatively high rate. Okay, so it produces kind of active tonic inhibition to kind of all your centers of voluntary movement. Okay, so the idea is an excitatory cortical motor command by itself will not cause a movement. Okay, you need to also pause the pallidum. Okay. And so let's look at the striatum. Neurons there are mostly silent, fire sparsely. As I said, inhibits the pallidum to release movement. Okay, so why do you want this convoluted seeming system? I'd say the basic idea is that it helps suppress unwanted movements. So you're not relying on neural activity being below a threshold. You're kind of actively inhibiting to just make sure you don't trigger unwanted movements. Okay, so now let's look at the striatum. So the interesting thing here, the main cell type in the striatum, medium spiny neurons, inhibitory neurons. And the interesting thing is they have a high density of this unusual potassium channel called inward rectifier. And so what that channel does is when the voltage on the membrane goes down, the channel opens. Okay, so it's kind of backwards of most channels. And it lets in a potassium current, which hyperpolarizes the cell even more. Okay, so it's a positive feedback mechanism that brings you into what's called a down state. Okay, right. So once you start to activate the channels, then they let a current go in that activates them more, and you go all the way down to like the reversal potential of potassium. Okay, so yeah, mention that. Maybe like minus 90 millivolts is the down state. So the important consequence, these neurons are often sitting in their down state. So then they have a really high threshold to fire. Okay. And you see this in the Rio base, which is kind of the level of constant current injection that causes at least one spike. These LAR5B pyramidal cells, it's, order 100 picoamps, and these medium spiny neurons could be almost 10. The data is pretty variable. Okay, so consequence of that at kind of the systems level is that it gives rise to a way of kind of simulating movement without doing it. So the idea is if the LAR5B cells are firing at kind of a normal moderate rate, what will happen is you still won't get a movement because you haven't activated the striatum yet. Okay, but the spikes still go down the pyramidal tract, and they trigger brainstem nuclei. They activate the thalamus. They kind of do all these other functions. And so the idea is that situation is one in which the brain is simulating what might be the consequences of making that movement. Why do that? So one thing is it allows you to integrate sensory evidence in favor of making that movement on longer time scales. So that's where the Thalamo-Cortical loops will come in. Another important function is to determine if your motor plan matches your goals. So the idea there is the cerebellum is forming kind of a forward model of what would happen if you did the movement. Then it feeds back to cortex, which can decide if it matches, and that would strengthen the firing in these neurons. But I'm not going to have time to talk about that pathway here. Okay, you know what? I'm gonna have to skip the global broadcast because I'm really running short on time. But just very briefly mention the pallidum also tonically inhibits regions of thalamus. So at the exact same time that you pause pallidum to release the movement, you're also going to fire another population of neurons in the thalamus that kind of spread signals all over the cortex. And so the basic idea is that communicates to the rest of the brain that you're doing this given movement. Okay. And there's kind of a beautiful paper from Ann Churchland, 2019, that shows direct evidence that this is happening all over the cortex, but I'm gonna have to So vote for that for.
I'm in retard.
time that's. Okay, good. Okay, so finally we get to a specific computational model. Okay, so the idea is, we've got these layer 5B neurons, they excite thalamus, they get feedback back from thalamus, they excite each other with some strength. And then the other kind of major feature here is they can get input onto their apical tuft, okay? And there's a whole bunch of experiments that have shown that this input raises the gain of firing of the neuron. Okay, so it has relatively little, it has some kind of depolarizing effect, but it also multiplicatively changes the gain of the neuron. Okay, so in this model, the idea is we have a pool of these layer 5B neurons. We're going to represent that with a single firing rate. That's actually kind of a particular subset of neurons that cluster together in their firing and encode a movement. Okay, so basic dynamics is you have a voltage variable that leaks. You get this firing rate feeds back onto the same population of neurons, and there's a parameter gamma for that strength. The thalamic activity feeds back with a parameter alpha, and there's some noise. And then the idea is the voltage variable gets rectified and turned into the firing rate by multiplying by the gain. So there's your gain control. So now these neurons in cortex, they deterministically fire neurons in the thalamus, but there's some kind of baseline firing of other neurons that puts a threshold there. So the firing of the thalamic neurons is firing of cortex minus the threshold rectified. Okay, so then, you can rearrange terms, and you have a very simple dynamical equation where this voltage variable, has this kind of this kind of effective decay omega and a noise term. Okay, so basically there are three regimes of dynamics depending on what that effective decay is. So if it's positive, when you turn on this additional gain, activity will rise to a steady state. Okay. If kind of the positive feedback balances the leak exactly, you get an integrator regime. And then if the positive feedback is higher, you get exponential amplification of behavior. Yeah.
Where did that rectification comes from?
The what?
The rectification of the input, where it comes from?
Just like the fact that you can't make negative action potentials. Okay, so now let's look at these kind of two loops competing. So the idea is, each loop has its own kind of amplification, and then they compete with inhibition. And so what basically happens is, whichever loop has like slightly stronger activity or slightly stronger amplification will win this competition. So this particular simulation is a case where properties of these two loops are the same, except this cortical gain is like 1% higher in one of the loops. So they both start, kind of integrating, but then the loop with slightly higher gain, it starts to go up more, but then that suppresses the weaker loop, and then that just kind of keeps going until you shut off the weaker loop. And then, another thing that kind of generically happens here is when you shut off the weaker loop, then you kind of you ramp at a higher rate, okay, because you've eliminated that source of inhibition in the thalamus. Yeah.
So where exactly the decision plays a role here? Are we talking about like spontaneous movements or task related movements?
We're talking about both. So this framework applies to both.
But the decision has to play some role to make a specific movements, right?
Yeah, I mean, so the idea is, let's say this Thalamo-Cortical loop encodes one particular movement, okay? And so without this cortical gain input, you're in this kind of damped regime where it's not a self-sustaining amplification. Okay, so these are variable amplifier loops. So basically, the key ingredient here is you need this descending input from somewhere else in the brain that turns up the gain on this loop in the context where you might want to make the movement. Okay.
So that's layer one?
Yeah.
Like how many loops will be participating in a competition at a time?
A little bit hard to say, but like potentially lots.
Okay.
Every sensory input is right, more or less.
Yeah, I mean, question?
So the competition's over like the sensory associations with the motor actions, right? Not over the motor actions.
That's one regime where, let's say, there's sensory evidence that is being integrated by different loops, and the evidence like in favor of activating that movement. And then they compete, and you're doing kind of integration of sensory evidence, but there are other factors that can also influence.
But you're doing that in kind of motor plant space, not in
Yeah.
senses. Yeah. So you very quickly convert to the motor world.
Yeah, so it's kind of a simple system where whichever loop has higher firing in the thalamus is gonna win. Like everything boils down to that. It's kind of simple.
It takes 6 seconds. That's an enormous amount of time.
This completely depends on parameters I put into the model, so you can make it much faster. But like the interesting thing is for it to be a longer time scale. So these parameters are long time scale integration.
What's
Yep. Go ahead.
what's the definition of a sensory input? Is it?
Well, it's
all vision,
sensory
or
inputs.
is it
It's kind of
something secondary?
sensory information that directly synapses onto these neurons. So like for instance, these layer 5 neurons, they get a lot of input from layer 2, 3. So let's say layer 2, 3 in cortex is encoding, some sensory information, and so the point is, then what you want to do, let's say for motor learning, is just learn synapses for that particular sensory information onto this existing Thalamo-Cortical loop that will make a particular movement, like lick left or something. Question back?
So you're basically a binary thing, so you either do the movement or you do not, and whichever move has highest activity kind of wins.
Yeah.
But like the firing rate of the population that wins doesn't matter qualitatively, like it only matters in the sense
It
that-
just has to hit a threshold. Yeah, and so the point is, like, that's the threshold that activates the striatum and releases the movement.
So is cells possibly in multiple loops simultaneously?
Okay, so
It's not clear to me
Yeah.
how you divide loops.
Yeah.
the way you're
So
talking.
the idea is there's a cluster of activity in the layer 5B pyramidal cells. So those clusters are, like in general, they're combinatorial. They're kind of, so it's a combination of certain neurons firing together and the rest of them being silent. So it's not like, so you have a lot more capacity than just like the number of neurons, because it is combinatorial. So one layer 5B cell could participate in multiple movements, but depending on what all the other cells are doing, there's a well-defined cluster that is moving A versus B. We could talk about.
So I want to push on this timescale because you are speaking at a very fast rate, one word after the next.
Yeah.
Now presumably, the plan of what you're going to say gets organized, but then, you have to move those muscles really rapidly.
Yeah, I mean, this, I could.
No, I understand that, but what it really means is that real time scale is probably sub-second. It's probably the millisecond range.
Well, I mean, yeah, so the issue is, like, if there happens to be just a very strong sensory input to this, group of layer 5B cells, then like you'll just, almost immediately reach threshold, get the movement, so all this, kind of Thalamo-Cortical loop dynamics, it comes into play much more when, it's not completely obvious from your sensory inputs that you should do a given movement, where you're kind of weighing different possibilities, you know, and that's the state we're in, you know, a lot of the time.
Okay, well, I think there's a distinction here, you mentioned voluntary. That requires cognitive decisions, right?
No, it's not cognitive.
No, no,
It's
no, I'm
just,
saying- I'm
it just
saying that
requires
that-
you shut off the palatum. That's what I mean.
Well,
by
no, no,
voluntary.
all of it's gonna work with the same machinery, but I'm just saying that there's a real difference between something that's extremely well practiced, which is, you know, what the basal ganglia is doing for you, in which case, you know, you trigger it and it just keeps going, right?
Yeah.
But there's also the voluntary, you know, cognitive decision, you're thinking, should I move or not?
Yeah.
Which is much, but you're saying the same machinery.
Yeah,
is for both.
it's the same machinery. So in the limit of like, you've learned this really well, you just have like a very strong sensor input here, and then boom, you get that movement. You know, and the loop doesn't even have to play much of a role.
But it's really, basal ganglia does sequences of movements, it's not just one movement.
I mean, cortex does too. I mean, you know, it's kind of, I would say it's a question like who's doing the sequences, you know, like personally, I think it's more of a cortical thing, you know, but you know, we could definitely discuss that. I mean, the thing about, you know, the striatal neurons is they have this very high threshold, they're really hard to fire, you know, the circuitry within basal ganglia is, the lateral circuitry is not that strong. So definitely plays a role, but yeah, what I mean, let's talk about that later. Okay, so I'd really like to get through a few more slides. Yeah.
Sorry.
Mhm.
You said some few slides earlier that each loop encodes a specific movement.
Yeah.
But movements could be very high level, you said, so it's somewhat hierarchical,
Mhm.
right? How could that possibly then do this winner take all?
Yeah.
You have many loops that should win, right?
Yeah. Okay, that's on a later slide, so let me defer the question and see. Hopefully I can get there. Okay, so just quickly mention the readiness potential. So this is like this very simple task where you're just sitting there and you're asked to like press a button whenever you feel like it. Okay, and when you measure EEG on people, what you find is there's this kind of ramping excitation for oftentimes seconds before you actually press the button. And there's been a lot of, you know, discussion about this in terms of free will, blah, 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 blah. But the main issue is like there are these kind of ramping dynamics of brain activity. It's mostly over frontal lobes. And so, yeah, so the basic issue is with these Thalamo-Cortical loops, you know, let's say they're in kind of an integrator regime and you just have spontaneous activity driving them, then, you know, the loops are gonna ramp. And another, you know, kind of characteristic feature of a lot of these data is that the ramping often steepens. So again, that's kind of a characteristic thing you get out of these competing loops. Okay, so kind of a very important experiment about evidence accumulation comes from the work of Mike Shadlin with this random dot motion task. So briefly, this is with monkeys. So the idea is the monkey is trained to fixate on a central spot, and you know, the researchers have figured out that there's a neuron in the cortex that has kind of a receptive field here. They've already identified. So then what happens is two targets appear, okay, and those are the two places that the monkey is supposed to saccade to. Then the motion stimulus appears, and it's a bunch of these like flickering dots, like dots appear, move for a few video frames, then disappear again. Okay, they're flickering, but a fraction of them are all moving in the same direction. Okay, and that fraction is called the coherence. And so if they're all moving in the same direction, you have a very strong percept of motion in that direction. But if like 1% of them are, it's kind of it's nebulous. So basically the task of the monkey is if the dots are moving to the right, then it's supposed to saccade to the right. Okay, and these different levels of coherence can make the task harder or easier. Yeah, coherence, fraction dots moving the same direction. Okay, so here's kind of basic data that they get from cortical area LIP. So on the left, this is the firing rate of a population of neurons they recorded relative to the time that the motion stimulus comes on. And what basically happens is you get kind of like ramping activity, and how fast it ramps depends on how strong the motion stimulus is. Okay, faster, stronger motion stimulus, really fast ramping, weak, etc. So now these are the same neurons, and they're triggering on the time of the saccade. And so what you see is, regardless of how fast the neurons were ramping, like leading up to the saccade, all their firing rates like fall into the same trajectory, and they all have a common firing rate, you know, kind of a or threshold for triggering the eye movement. And then the other thing you'll notice is there's like a certain amount of ramping here, but then in this regime right before the eye movement, it actually the ramping gets steeper.
These are averages, it's not single trial examples.
These are averages over like of order 100 neurons in many trials. That's right. Okay, so in this model of Thalamo-Cortical loops, you know, the way you can kind of model this behavior is you've got this motion signal here, kind of left or right. This is kind of a Gaussian variable in this case. The idea is you rectify, you know, the leftward motion and the rightward motion to two different channels, and this is, you know, what's seen in cortical area MT, like the neurons are direction selective, you know, left or right. And then basically you connect the leftward motion neurons to you know, a Thalamo-Cortical loop that will do a saccade left, and the rightward motion signal goes to saccade right. Okay, so now if you run the model, you know, what happens at a very low motion strength is like the left and right Thalamo-Cortical loops, you know, both ramp up, but it's pretty noisy, and sometimes, you know, the wrong direction wins. But at higher coherence, the kind of the correct direction, you know, ramps much faster, so you reach threshold sooner, and you also are correct more of the time. And you know, so qualitatively, that's what you see in the experimental data. This is probably being correct as a function of coherence. You know, at sufficiently low coherence, it's close to 50-50, and it kind of sigmoidally goes up to 100%. And then movement time also kind of systematically decreases as you increase coherence, and that's kind of qualitative agreement with the data.
So
yeah.
So the student was basically said it's one of variable, and you actually change the mean.
Yeah, that's right. Yeah. So in that case, it was always the same standard deviation with the mean. Yeah. Okay, I'm gonna skip this in the interest of time, but you can actually compare this model of Thalamo-Cortical loops directly with the drift diffusion model of decision making, and you know, it agrees very closely, and the Thalamo-Cortical loops model still approximates it quite well, even if you mistune the parameters of a fair amount. Okay, so here's like the last main point I want to get to. So in this gets to your question about the hierarchy. So actually, you know, in this motion task, there are two different versions. The one I described is the reaction time version, where as soon as the monkey feels confident that it knows the motion direction, it's allowed to move its eyes. But another condition that you know Shadman lab studied is a fixed viewing duration. So the idea here is the motion stimulus is on for like 1 second, no matter what, and the animal is not allowed to move its eyes. And then it turns off, and there's like another like 1 second delay where the animal has to kind of remember what it wants to do and not move its eyes. And then there's a go cue, which in this case is the fixation spot disappears. Okay, and that visual stimulus tells the animal to make the saccade. And these sort of delay tasks are actually very, very common in neuroscience. You know, so the question is, you know, in this TC loops model, like once you reach this threshold, you get the movement. So like, how can you suppress your movement during the delay period? And the answer is with a hierarchy of Thalamo-Cortical loops. So the idea is here, these are the loops we're considering that are, you know, looking at left motion versus right motion. And we have this like cortical gain coming down. Where does it come from? Well, it comes from somewhere else in cortex. Okay, so the idea is there's there are loops at a higher level of cortex that essentially like decide we're going to do the task now. And the firing rate output of those neurons goes down the hierarchy, and that is the gain variable to the lower loops that are doing the evidence accumulation. Okay, so the idea is there is kind of a competition among a bunch of loops at the higher level, like in prefrontal cortex, to determine like what are you going to do now. In this model, there's kind of an increased gain to the loop due to like task-related factors. So in this case, it would be those fixation targets appear on the screen. So that provides kind of input to layer one of these neurons in let's say prefrontal cortex. And so there's going to be a sustained gain as long as those fixation targets are there. And then the other piece is that the go cue is going to give you extra synaptic, extra stimulation of the higher level loop. Okay, so what happens for the higher level loop is like you turn on the gain to do the task and you get, you know, kind of an elevated activity that is sustained. And then when you get the go cue, you get an extra bump of higher activity. Okay, so then this is exactly the signal that is the gain input to the competing loops. Okay, so now let's say this gain puts you roughly in the integrator regime. So when the targets are present but no stimulus, like there's no integration because there's no motion stimulus. When you turn the motion stimulus on, these two competing loops integrate motion evidence, and you know, let's say one of them has more evidence, so it goes up to higher firing rate. Then during the delay period, there's no more stimulus. These two loops still compete, you know, and so this loop with a higher activity, it roughly keeps it actually suppresses the lower activity a bit more. And then what happens when you get the go cue is that the gain goes up, okay? And so that puts the loops in this exponentially amplifying regime where, you know, whichever loop is higher just immediately wins the competition and generates the movement. Question?
So this is also where you say like a sequence would then be generated in the higher loops where you kind of prime all these different sequences and then just fire them off.
Yeah, here you're priming them to put them in the regime to integrate sensory evidence.
Right.
And then when you get this extra bump of activity, that's gonna tell them to like make your decision now.
Yeah, but you're saying that there's no sequence, there's no understanding of sequence in the lower cortex, right? You'd have to have some kind of higher control to sort that.
Well, I mean, so what I'm saying is if you just had these loops in the lower cortex, as soon as you had enough evidence, you would make the movement.
Yeah.
You know, yeah, and monkeys can do that, of course, but you can also make them do this delay task. You know, so the idea is, you know, in the delay task, you have this kind of important role of let's say a higher cortical area that, you know, primes the loops to do their ramping and then triggers them to go into the exponentially amplifying regime to make their decision. Okay, so summary, single Thalamo-Cortical loops are these variable amplifier circuits in this view. So the gain is controlled by descending cortical input, so you know it's tunable. And kind of a useful regime here is to have a gain that tunes the positive feedback to roughly match the leak, and then you get kind of linear ramping for spontaneous input. So that qualitatively agrees with ramping activity, neurons, and readiness potential. Multiple loops, you have a winner-take-all competition. So that gives you this kind of global motor coordination where only one movement can be driven. We integrate sensor inputs. That's also like the drift diffusion model, decision making. And then the global broadcast, I didn't really have much time to talk about.
Wait. There was a word that was flashed.
Yeah. Okay, so there's the global broadcast has a lot of features that are consistent with being the neural basis of consciousness. So happy to talk about that later, but that's a different talk. Yeah. Question?
Sounds good, take it away.
Okay, thanks. Thanks, sir. Okay, so I'm giving this talk about curing the Silicon Cochleas. Okay, so basically it's about my the work that led to this prize that I got, the Mishamahawa Prize back in 2020. And I just show you the team members. So basically I had a bunch of students who work on algorithm, circuit design, and also signal processing. And actually, I think a bunch of developments which I think were plugged for the field. And so I want to talk a bit about this journey of all the work that we did over the last probably now about 10 years. And so let's talk about the history of neuromorphic curing. And so to put everybody in the same place, so I'll give you a little kind of a description of the anatomy. So basically sound is coming to the outer ear, goes to the channel, then hits the eardrum and causes the bones in the middle ear to vibrate, and which then this vibration is then going to the inner ear, and if you unroll the cochlea, so this is the hearing out. And what you see in there is this is a simplified picture. You see a piece of membrane that sits in the fluid chamber, and the membrane is shaped in a way such that it's going to pick up different frequencies at different places along the membrane. And so there are already experiments done back in the 1960s by Casey, where he measured the displacement of the membrane as a function of frequency. And so you can see that if you're very close to the states, the last bone in the middle ear, then you're sensitive to high frequencies. And then as you go along the membrane, then you're sensitive to low frequencies, right? And if you are an electronics person, then you look at this picture, you say this is a band pass. So I can build an electronic model of the band pass. And so this is kind of the model that Carver Mead and Dick Lyon built, right? It's electronic model of the cochlea. And this was something they already did back in, you can see the date of this article that they wrote. So it was
it's too small.
that.
What is their open number?
Yeah, so this article was published in 1988, right? It's called like Analog Electronic Cochlea. And here's the layout of your chip given by Dick Lyon. And you can see that even back then, you already built a few hundred tabs or filter channels. Now, on top of this vascular membrane, actually sits a bunch of cells. And so there are two different types. There are the inner hair cells and outer hair cells. And so you can see them. So here's the vascular membrane. And so actually along here, you have partitions. And each of these partitions, you would have one inner hair cell and 3 outer hair cells. And then the output of the cells basically form these fibers that go out. Sometimes what people call the auditory nerve fibers. And so you can see here on this hair cells that if the whole cochlea vibrates, then what happens is this hairs on the cells will then start bending, right? And that allows the ions to flow in. If it bends in one direction, and then it doesn't let ions flow in, as you go in the opposite direction. And then when ions flow in, then it causes spikes to travel along the pipe. And so you can look at recordings like this, usually from the cat at the time back in the early 2000s. And you can see kind of the spikes that come along the fibers when the animal hears sounds. Now, of course, subsequent to the work that Carver and Dick Lyon did, there were a bunch of students in Carver Mead's lab that work on better and better silicon models of the cochlea. So there's Rahul Sapeshka, Lloyd Watts, across in Switzerland, Eric Vitals, who was also writing a similar neuromorphic work at the time. So Andrei Van Schaik was one of his students, and he also worked on a silicon model of the electronic cochlea. Here you recognize the younger Popena. And then subsequently, his silicon ball when yes, well, right. You developed a nice, much more faithful model of the cochlea. And a lot of the blocks kind of look like this, right? So you're going to have a set of filter channels, right? And what you can choose to do is to build it up to here, so you only have the filters. Or you can build also the model of the inner hair cells. And so usually we model as a simple halfway rectifier. That's why the hair is going to bend one way, then it's flowing, otherwise it doesn't. And then you can put a very simple model of an integrated fire neuron, IEF. right? And then you get spikes coming out, and then you transmit the spikes using AER, this address band representation, similar to the DBS, right? And then you can use it just like the DBS. So, sorry, what are they coding the spikes in every hair movement? Sorry, what are the spikes in coding exactly? The hair movement? Oh no, so what the inner cells do is, because they rectify the signal, they actually measure the power in the frequency channel. Yeah. And now this, in some of the silicon chips, people actually do a full rectifier to have the rectifier. And so this is just to give an idea of the kind of the number of silicon cochlea designs from starting from the one that Giulia and Carolini did back in 1988, right? And this comes from one of the chapters in the book where Tara Hamilton, a student of Andre Van Schaick, actually went and collected kind of all these different publications and then made like a historical tree. And so if you're interested, then you can kind of just glance through this, I don't like a family tree of cochleas, right? And you can see that people have different ways of implementing the circuits for the cochleas. So sometimes, so in this first one by Giulia and Carolini, so the filters are actually cascaded. It's a cascaded architecture. So the output of the first filter goes to the second filter and so on. And the reason for that is that you can get this very steep roll-off that you want in the filter, right? So it matches the biology better. But subsequently, there are other designs where people build like a parallel filter bank. So you take like the input coming in, and then you feed it to the filters all in parallel. And also then there's different ways of designing the filter in silicon. So sometimes you can do it with straight analog circuits, with transistors. Sometimes people use switch connect, right? And then there was this lovely work that came out under Van Schaick's lab, where they figured out a way of reducing the mismatch in the filters, because usually, you know, with analog circuits, you get mismatch. And so what happens is this beautiful type of impression. These curves that you see here, you can get something similar, but if you don't deal with mismatch properly, then you'll find that one of your channels will have this gain here, another channel will have it down here, right? Maybe it's broader, and another one might get it higher, and so on. So you really have to think about the circuits like you're designing. And then subsequently in 2014, together with Andre Van Schaick and Toby, we designed binaural cochlea, which we subsequently called the dynamic audio sensor, like the dynamic vision sensor. It's called a DAS. And so you can see now this chip is actually sitting here, and there's a printed circuit board and all the components that support the chip. And also Toby wrote this Java program JAER, where you can then read off the spikes in real time, right? And so the chip has 64 frequency channels, binaural cochlea, spiking outputs using AER. And here's a recording from the channels, right, which are basically spaced from the center frequency is from 8 Hertz up to 20 kilohertz. And what you're going to see here are like master plots, and they color 2 different colors for the 2 different years. And you're going to hear a very nice sound, and you can see what happens. Go up, then different channels will start spiking. Then you can see also the red and green, and when the spice level at the time, then you see yellow. Also red and green are the 2 channels. Yeah, red and green from the 7 channels, and then when you overlap, continue the left and right. Yes, the left and right, yeah, whichever left and right. So you can choose the. Oh, sorry. Okay, and then we had this lovely student, Min Hao Yan, who oh, sorry, she didn't touch the board. That was really interested in getting the circuits designed so that you can send it to, you know, this conferences. ISNCC is like the primary conference for solid-state circuits. People really try to get your papers in there. And he really did a lovely design where first he decided that he wanted to do a parallel architecture. And then he took the output of the filter and then he passed it to an asynchronous delta modulation. And so now it's actually the design is a full wave rectifier, so you can actually get the positive and the negative side. And then he sent off the spikes or the events as the on and off events, right? Similar to the DVS. Right? In the real cochlea, you actually just get one side. Right? He said, look, just do both sides, then they're both similar. And then we try to do something with the output of the chip. And the beautiful thing is that if you look across the 64 channels, right, of 1 year only, the other year it looks the same. You can see how well they're matched, right? So the gain is all around the same place. The roll-off, all kind of, really like each other. And if you set it to a Q of 10, so Q defines the quality of the filter, so basically defines the sharpness. So the higher the value, the sharper the filter. And so now if you play a chirp like I did before, then you can here see the output spikes of the channels, right? So this is channel 60, which is sensitive for low frequencies. And then you can see as the frequency of the sound increases, right, up to 21 kilohertz, you can see how tight this line is. Like if it's not so selective, then you're gonna see like this broad horizontal bars. So I think that's a really beautiful design that they cut. And the lovely thing that you also did with this design was that it burns only 55 microwatts. And it's extremely low power. And so for the technology that we were using, it was also quite impressive. And the good matching across channels. And so that you have an idea of what this conferences look for, right, is that you usually have to supply like a comparison table. And then you have to have metrics here on the side, right? So for example, you have architecture, technology, your supply voltage for your chip, number of channels that you have, a frequency range of your channels, the Q, the power, normalized to a particular frequency channel in microwatts. Then of course, area is always important, area per channel, and then the dynamic range of your sensor, and also whether it's binaural or not. And so anyway, we put together this table along with the other cochleas that were designed, or a few of the cochleas that were designed over the years. And so here's the one that we did for the DAS, and then this is the one that came out for Ennis group. This came out Eric Vitus's group, and from Rahul, and this was the design that I just showed. And so you can see how the architecture is different across the different designs. You have cascaded, coupling, passive, parallel. And then the technology is kind of similar. 0.35 to 0.25, okay, this is a very old technology. And then the supply voltage, you can see for Minha's chip, he ran the design at 0.5 volts, right? Versus just other designs that go between 2.5 and 3.3. And why is that important? That's because the power is dependent on your voltage. Like the lower you can bring down the voltage, the power supply voltage, and your circuit still works, the lower will be your power, right? So that's something that will value kind of low power designs. What else should I point out here? Okay, I guess here are the other main things. So the Q range, you can tune the design from 1.3 to 39, which is pretty high quality factor. And then also the lowest power design, normalized. Yeah. Okay, and then of course, something that you have to sacrifice to get the mismatch down is that you have to increase the area of the circuits, right? Because that's one way to bring up mismatch. And then finally, you also had the highest dynamic range of out of designs except for this one. Yeah. Okay. Any questions? Because I'm trying to give you guys the impression of, what this is look for, right? And also what you would like to think about in your designs if you're doing something like this.
What is the frequency range for like human hearing?
For human hearing, it's somewhere between 100 to 20 kilohertz. Yeah. And actually, if you're doing a design for a certain application, this range is too broad. I You don't need such a wide range. And when we give talks, they always say, why do you have to do this? Because they don't care about building the human coping. Solve the problem. Yes.
The output of the system, where do you intend to go, or how do you intend it to be?
Oh, I think they go with the chip.
Yeah.
I'm going to show you all that stuff that, they with the output.
Looks like the power is almost like whatever is society. It's not all six place all that, or whatever other choices.
Yeah, also, of course, other choices, circuits.
Yeah.
Yeah.
Yeah, you just, are you going to talk about the circuit?
Yeah, not to talk about the circuits because I have 1 hour. This talk I'm giving, you can have like 2 talks from there because like can discuss it separately. I mean, we wrote actually I had this lovely postdoc in my group, Quantic Kim. I traced some one of the designs we have, the most recent one, and we wrote an article in the Circuit and Systems magazine, and we gave the review of all the different circuits that people have come out with the filters, and then we explain. Right. Yes. Yeah. So if you're interested in this kind of the circuits for the filters, just look at that paper.
These are all spiking, not right.
Yeah. So all of this put out spikes. Yeah. But the subsequent ones that you'll see that we don't always put out spikes because we combine it with the network on the same chip. So we don't have to send the spikes on. Yeah. So this is kind of the pure sensor. Questions?
So getting down to 8 hertz is very impressive.
Getting down to,
I mean, his low frequency is 8 hertz.
Yeah. Yeah. That's why this chip made it to the conference. They were already impressed with this. The fact that you can run the circuits at 0.5 volts, right? 0.18 microparsecs here.
So you use that big gap there for 8 hertz, or some kind of?
Yes, the area is big, but this chip is big. Yeah.
So that's kind of a question. I mean, if like a 10 or 20 kilohertz chip, so this chip is going to be putting out spikes at like 2 kilohertz.
Yeah, did you design the chips to like this? Yeah, I mean, he's in the designs that we did later on. I mean, the one that I did, we did like fixed voltage, right? So you're right. I mean, as the frequency goes up, then you're going to get more spikes for the time. Yeah. With the delta modulation, it's safe. Yeah, it starts. You can always low pass filter first, right? Before you get out the spikes. Like, if you want to know just, kind of the ID, well, because which channel, right, sensitive to high frequencies, then you can just put up spikes here, channel know more about this market. Yeah.
So this spike is delta modulator or immediate fire?
So for the different designs we did, they sort of vary. Yeah. So this one is, wait, let me see. So the one that we sent, this one is delta modulation. Yeah. The one that I did was integrated fire. Yeah. Subsequently, when he left up and became a postdoc at Columbia, he did one with integrated fire. Yeah. Okay, so just again, it's another picture where you have the 64 channels and then the spikes will outrun the timing to the board. Yeah. And so I don't remember what the speech was, but you get the sense of kind of spikes flowing through when there's sound, and otherwise it's completely quiet. And also you can take the spikes and then build a couplet one, right? So what that means is you take the spikes and then you choose a certain thing time. And in this case, it's 5 milliseconds. And then you basically plot how many spikes you get per frame, per 5 millisecond frame. And then for this particular sentence here, shaded ducks with receive wash water all year round from one of the data sets that people use a lot, right? Then you can look at the input spectrogram, right? And then you look at the cockpit lab. And then basically you want to be able to show that they have similar patterns. And so, yeah, you can kind of see that, right? Like when you have power over here at the lower frequencies, you also see the same over here. So it's like the information is there from the spikes of the sensor. And so anyway, to further prove that you can actually get back the original sound, doesn't mean that you have to. So what we did was we took the output spikes of this particular sentence, general plate 9 straight. She had a dark suit and greasy wash water all year. And now this is the leafy stripe. So she had a dark suit and greasy wash water all year. Yeah, so you can get back the audio because it's not as high quality as the original, but yeah, the information's all there. Okay. She had a dark suit and greasy. All right, so another thing that we did with the spikes was that we did localization, right? And because remember, the spikes carry timing information, and this is a very good for a test of localization because we can use these cues. So normally we use these cues up into our time difference and into our level differences, right? And so if you want to know where sound source is, if you're in the middle, then the time between the sound getting to all peers is 0. And as you go across here, it's about 700 microseconds or so. The sound, so here, if you go, it's out of the starting in microseconds. And so what I wanted to do is to see whether if you take the spikes coming from the left and right here, and then can we compute the delta t, right? So that we know where that sound source is. Of course, one thing that we always have to deal with is that this is not a simulation. So if you look at the output of the chip, you can see from one here, you get this red spikes, right? For the different frequency channels, these are the different rows. And then for the same sound, you get the blue spike on the other ear. And you can see the spikes don't, it's not 1-to-1, right? So every time I get a red spike, I don't always get a blue spike and vice versa. So then can you really extract this delta t, which is the time difference between the red spike and the blue spike, right? And then create some sort of distribution. And that's what we did here. So you can see a picture where this is delta t of 0, and then plus minus something. And then basically what you do is you compute the delta t's across all the spikes, and then you build this distribution, you look for the peak, and you say, notice where the sound is. And so what you're going to see now is a video where we have a setup with both the DBS and the DAS on the same platform. And you see here the output spikes in the cochlea, the output of the DBS. And then this is the IP. So you're being continuously, you're doing a concept detection. It's continuously, continuously, yeah.
You will see the spikes from the cochlea and a localization result showing the peak where the sound is located. With this, the control unit is driven such that it orients towards movements in the visual field and towards the localized sound.
And so if I now start talking, let's see if it moves towards me, and yes, it does.
And now I will start talking, and now it moves back where I am.
Right. Like that. So you can see that you can do the localization in my messy office.
But Shih-Chii, you want to say something about the main thing that really made it work to deal with reflected sounds and noise?
Yeah, I mean, so the algorithm, so it's not, has a better memory than I do. Okay, so it's not just enough to compute the delta t, right? And so, you know, when you're in equity room, just using delta t alone, it's not going to be, it's not going to give you precise information because you can get this reflections, right? And these reflections can lead to incorrect delta t's. Right, and so what we did was that we always do a weighting of the delta t before a pause, because if there's a pause or a silence period, then you know that's the direct sound coming at you, and then in between it's not the direct sound anymore. So if you have like a long time before you compute the delta t, then you weight those values higher. That helps.
How long is a long time?
So long is a long time. Yeah, I mean, it depends on the road that you're. So I don't know, like I don't remember. Yeah, I don't remember.
But what
it's in a millisecond range.
But you see what the onset detection would help here. The onset
it is an onset detection.
I saw an onset detection, right? Because of silence. So it's just so then you can run the sound, you know, basically run the algorithm all the time.
Yes.
Yeah.
You always have pairwise.
Yes.
Place to do something.
Yeah, we always do pairwise, and so we look. So every time I get a spike, I look across to the other side, right? And we only pick the delta t that's within this range that I just mentioned. So if it's outside of the range, because the other side didn't fire, so then just say, okay, so it all. But this for all channels, Yeah, we do it across all channels.
So that's the nice thing about this compared to cross correlation. Objects are very cheap.
Yeah.
Because the delta T accumulated into ancestor is challenging it's delta T's and L's.
Yeah, also the spikes here from this from the jazz was spacewalk. So that's also very useful. Yeah, you can do it with the delta modulation outputs. I think you like that answer. It's not it. Okay. Okay, and then so we did this work already. And then subsequently we did this other work with your Conrad, which some of you might know. This group built this lovely pen-tilt head, and so the idea was that we could have stereo DBS cameras, and then we could have a neck, and so you can do like this, like your head, and you can do your eyes like this also, right? And then the microphones are mounted on the sides, and then they go out to the pocket board, which you don't see here, right? And Philip Klein did this experiment. He was a master's student at the time in a group, and you can see the platform here, so you can see the DBS, right? And then the microphones are here on the side. And on this part here, you see the output of the two DBS cameras. And then here we do scene stitching, right? Because the DBS can look about, I think here about 60 degrees. And so to get the whole scene, right, you would have to do this panel, right? So my shutting to stop making. Yeah, so you can see it's panning around this way, right? And if you look carefully, you can see that the camera's are pointing down, and as it turns around and goes like that, it starts moving up, right? And so you can see now, you see the scene is being stitched together. Yeah, and so by the end, you see the cameras on the, so you can get the X and Y output. And that's the final picture here, which will be hard to see. I mean, if you didn't see the original, you couldn't see it. I don't remember. I think these are the chairs sitting in front of a blackboard, and on the blackboard, he drew a face here, and then like the little house. And then you can do this thing of like selective visual attention. You could like pick out the person who's talking, and then you paint that person, right? And so the experiment setup has a speaker. So that's supposed to be the person speaking, right? Oh, sorry, that's the person speaking here, here, or here, right? And then I forgot which way does it go. Okay, right. So you stand here, right? So that the cochlea knows where you are because you're speaking, right? You paint the person from when they're in that position, and then you can create like a scene like this where that person now is in these different parts of your own picture, right? So you see he moved from a particular time at 30 seconds, he put himself here, and he moved over to a different spot, and he kept on talking, moved to another spot, kept on talking, and moved to another spot, kept on talking. Right, and then you can paint this picture.
But there was a 500 hertz shown also. It's another source.
Yeah, so the 500 hertz, I don't remember why he had the 500 hertz and why it was in his picture, to be honest. I think initially when he tested the system, he moved the speaker around, right? The speaker, not the person, right? Just to make sure that the system is working properly. Okay, so so there are many questions about why use the cochlea with this asynchronous output versus just using uniform sampling. And so this is something that we try to quantify, right, in localization, especially because that timing is useful. And so this is a picture from a CAS magazine article. And so here we plot the localization accuracy as a function of the time or the latency of when you want to put out the localization information. And so you'll see two groups. This is the uniform sampling group. So this is when you uniformly sample the sound coming in, right? And you do the cross correlation as Toby says, right? To figure out where the sound source is. And then you can also do event sampling. So this is using the events of the cochlea, right? And then you can choose different frame times before you report the localization output. Like so 8 milliseconds, 1632, 64, 128. And then at the same time, we also computed how many flops you need, right? For this different parameters, okay? And so if you look first at the uniforms, okay, actually you should look at the event sampling side first, right? So first you can see in general, the circles are smaller. So basically it's cheaper, right, to compute localization versus if you do a cross correlation. And also the localization accuracy is higher. And if you look and compare with the uniform sampling case. But if you allow enough time, if you allow the latency to be longer or the frame time to be bigger, then you're going to actually get better localization accuracy using the uniform sampling. So it really depends on what you're looking for. If you're looking for like a cheap way to get some estimate of where the sound source is, then you can get it from the event sampling method or the spikes. Otherwise, if you really want to be precise, then you should use the conventional method. So I think this is a nice outcome because to me, the conventional method does give you very good results. And so if we want to claim the event sampling method somehow has some magic where you can do fewer computes and still get the accuracy, I think it's very difficult to show this. Yeah, I wouldn't know how to show it. So anyway, this is a summary of what we understood from the localization work. And then we also did some work where we did real-time speaker identification from the cochlea spikes. So what we did here was we extracted features again from the DAS. Back then we call it AURDR2 before we call it DAS. And then we took the feature vectors, and this was prior deep networks. So we classified it using a linear support vector machine. And then we used a data set from this timid database. So actually it's a subset of 40 speakers from timid with 10 sensors per speaker. And then we look to see how well we can identify the speakers, where they correctly identified more than 90% accuracy and a latency of 700 milliseconds if you switch speakers, right? So basically you have one speaker talking, you identify which of the speakers it is out of the 40 speakers, and then you switch speakers like, just suddenly, and then you see how well it takes for the system to recognize that it's a new speaker and it's the correct speaker. And then for the feature vectors, what we did was we had a frame time of 15 milliseconds. We did two things. So one is we took the spike count from the different channels, and this is one feature. And then the other one that we used was the average interspike interval distribution across all the channels. And then those are our two feature vectors.
So why did you have horizontal 8 output neurons? You said 5 will.
Oh yeah, in this design that I did, I actually had 4 neurons per channel because I wanted them to have different thresholds.
What did you just to have Linux?
Because this is what you have also in the real cochlea. So you put in everything the real cochlea has, and then you figure out which ones you don't really need to solve the problem. You start taking the account. Yeah, but this one we had. Yeah, this was like. And this is so polished. Oh yeah. So what you're gonna see here is okay. So these are your 40 speakers, and then you're gonna hear sensors uttered by the different speakers, right? And the red bar basically says if it caught up or not, or it's on the right speaker that's speaking at that moment.
Will you please confirm government policy regarding waste removal?
The meeting is now adjourned. The dark, murky lagoon wound around 4 miles.
An adult male baboon's teeth are not suitable for eating shellfish.
I'll have a scoop of that exotic purple and turquoise sherbet. The frightened child was gently subdued by his big brother.
The speech symposium might begin Monday.
Did Sean catch that big goose without help? The dark, murky lagoon.
Okay, so you guys get the idea. is that, you know, when one of them is talking, the red bar, if you pick the right speaker, it'll be on top of it, right? And then you can see how fast it switched to the next speaker and so on. Yeah.
Will you please?
Okay. So now I'm going to move to the work that we're doing in recent years, which is using deep networks and trying to solve a particular problem. Right? So moving a little bit away from like just keeping the features from the biology that's useful. And so the reason for that is because the edge audio market has become very important. Like we look at the, you know, the amount of revenue you would get out of this market, presumably it's going to increase by 2x, 27 billion by 2026. And that's because a lot of these devices that we have nowadays are voice activated, right? So it's a very important thing, very important, critical. It's a very important component of any one of these devices. And of course, since all these devices are portable, there's certain specs that are also critical. So first, you want your device always on. You want to do some local processing because you want to send limited information to the cloud. Also, you want it to respond in real time, and it needs to be small. And all these properties basically places a constraint on the power and also the memory that you can have on your device. And the usual way that people do this currently, not just in the neuromorphic world, but also in the more conventional circuits world, is that you have a front-end feature extractor, then you have a classifier behind it. Usually it's a deep network of some type. And then you co-design it so that everything fits into one ASIC. Yeah. So basically you don't have like a chip that's only a sensor output that clicks out only the output of the sensor. Okay, and so in our case here with the Cochlea still inspired, so we have a set of analog bandpass filters going to a rectifier and then creating events, or you can take the output of the filters and then create events from it directly. And then for the classifier, there are different architectures that you can try. You can use recurrent neural networks, or you can use a straight NLP.
Question the question. So automatically feeding this spike output, or you're trying to get the energy first, like low pass filter and then this spike?
Well, we've done it two different ways. Yeah, actually most of the time we rectify it because we want to get the energy first.
So you plug it there, get under X.
Yeah, exactly. And then you get the events that come out, and then you do like a spike count. So it depends on how you run the network, right? So if you run the network in a synchronous manner, then you say, okay, this is much time to collect the spikes, and then I test it in your state vector. Yeah. And for the kind of tasks that people look at is either voice activity detection, keyword spotting, or speech enhancement, because these are usually very simple networks, and you can put them on one chip. Okay. The other thing that we wanted to do is to compare ourselves against the conventional way of generating features, which is to first take to do an analog digital conversion of your microphone input and then pass it to a fast Fourier transform block, and then do some additional digital filtering afterwards. For example, if you want to get MFCC features, then you can also do that. And then now, in this case over here, which we call the bio-inspired or neuromorphic feature extraction, then basically you do away with the ADC, and then you just have the circuits for that thing. And so the thing that we're trying to show here is that, of course, you can save energy or power right away, because one of the biggest power components of this pipeline is the ADC cell, right? And so if you can get rid of the ADC, right, then you can save the power from that. And then the other thing that we wanted to also, at least for me, I think we don't have to be necessarily using always a SNN behind, you know, this feature extraction, right? So we can take any one of these guys, these different options, and we can feed them into a synchronous DNN, or you can feed into a asynchronous SNN, right? I don't want to say it's one or the other. Because you see that's a lot of interesting work that's also going on in this part, right? And that has brought them to very good power numbers compared to this part, right? So we have to look and see what they're doing, and they're inspired also like what we're doing, right? And so I think basically it's a win-win for both sides. Okay, so here, this is from the slide. I test all this. I think you guys know why people like SNNs, right? Because you don't have to do multiplication and summation. And then if you can guarantee that the events you produce are sparse, then you possibly could have high energy efficient systems. And then nowadays, there are very good training methods, deep network training methods that can lead to test accuracy, so they can also run the SNN behind it so that we can get good accuracy. And last thing is that we also a bit more open. We say the circuits that we build can be mixed signal or digital, like for ourselves. Okay, but one thing that is clear is that if you do asynchronous SNNs, right, memory access remains tricky. Okay, because you're still going to have to use synchronous memory components off chip if your network is stable.
So I have maybe dumb question. So yes, output from your copy and it's coming this delta mod list, right?
Yeah.
And then if you use this SNN, which typically use integrated file, right? So there would be compatibility issues, right? The kind of encoding you have.
Yes.
How do you manage that?
Well, that's why we don't really do SNN. So like, well, this is get people doing it. So, you know, we just chose to do something slightly different, but still inspired by the SNN. So we said is, look, you know, the usual way that people update ANNs, right? Synchronous ANNs is that you take the activations of the neurons, fetch the weights, and then you update the next layer and so on, right? But we could be like SNN-like, but still using the synchronous ANN framework, is that we choose a subset of the neurons to be active. There are some conditions where these neurons are active, and then you only have to fetch the weights of those neurons, update the neurons, the next layer, right? Apply the condition away again, right? Maybe this guy is the only one that's active, and then you update, right? And so there's a threshold involved in this, determining which ones are active. And so this is something that we call the delta network model, right? We focus mostly on recurrent neural networks rather than CNNs, but you can also apply it to CNNs. And then we train the ANNs for this sparse delta activations, as you see now.
Or I'm not sure with CNN.
What's that?
I'm not sure with the delta things would be a good option for CNN, right?
No, it's not
useful, right? You don't get that advantage to getting the RNN.
Yeah, but I think if you're doing like current CNNs,
recurrency,
yes, then you would have to start a state in
recurrency.
Yeah, there's some work going on in that direction. Yeah. And so we call this the event-driven temporal sparsity network. So you're temporally sparse across time. And also it's because we're using the synchronous updates, the memory access is also more efficient. I just briefly, I give you an idea of how it works because I think some of you already know this delta network model. And Toby said you would talk more about it in some stuff.
In touch, or because this is drugs. But
yeah, I was going to talk more about the keyword spot. Anyway, so this is just a typical pipeline, right? You get an input, you extract features, and run it to a network, post-process it, get the output. But then in this deep network thing over here, what we do is to run the delta network. And so at a particular layer, what you do is you store the state, right, at a certain time step, which is this level that you have here, previous to it. So give 4 examples. And then in the next time step, when you update the activation, then you see whether it's above the previous one by some threshold that you define. Yeah, so in this one, we call it activated because it's above. Like the threshold here, it's unactivated. And this is the other way. So you can also drop below the previous value. Right, so it's activated, and this one's not. So, and now you can see these are the 2 guys that update the neurons to the next layer, right? And then you go on that. Okay, and then to show that it actually works in real life too, is that we have this Cochlea chip. Here it is. Here again, you recognize it still, the 2 microphones. And then the Delta network is actually running on an FPGA, right? And here we train the network so they recognize digits. And so what you see here again are the spikes from the Cochlear channels. And then on here, you have the different digits. So this is 1 to 0 and then 0. Also has the labels. These are the labels. And then we also print it over here so that it's easier to see what the network detected. And this is Chen Gao, who did a lot of the work in implementing the Delta networks onto FPGA and showing that this idea actually works in real world.
2, 3, 4, 5, 6, 7, 8, 9, 0, 0, 0, 5.
Okay, all right, just to keep your mind. Yes. Yeah. Okay, but kind of clunky to have 2 boards, chip each and whatever. So later on you'll see that we also do an ASIC. We both things on the same chip. But in the meantime, we also wanted to quantify kind of the value of this space. If you combine this temporal sparsity, spatial sparsity, and this was done on again another FPGA platform, which we call Chen call it Spartas. And so what he did was he trained a layer of 1024 LSTM neurons on the 2 data set at 1. It's implemented on the Zing 7100, and then you look at the numbers from batch 1 info. So basically, you feed every sample, and then you see how it checks get the output. And so if you look at the savings, so this is energy efficiency, which is the units are giga operations per second per watt, right? And then you look at the efficiency numbers when the networks are pruned, which means that you don't try to take away weights, And if you now prune the network, so this is weight sparsity, you can see your energy efficiency could increase by the effects. And then if you add now also temporal sparsity, you can get an extra edge. Right? So all these different sparsity methods that you impose onto the network can help increase the energy efficiency of the system.
Yeah, I thought make a comment here because this doesn't look like a big gain, a factor of 3 from temporal sparsity. But you have to realize it's tested on a dense data set. In other words, this data set is continuously making sound all the time. So it doesn't represent real world sound.
Yeah.
Most of the time you don't have sound, right? Or the sound is stable. And the states of the network won't change if they're hearing a washing machine. The hidden states in the network just won't be changing it. They just hear the washing machine over and over or something like that. So it's a little bit, this is like the worst case.
There's always sound.
From temporal sparsity.
Because you're saying that sound running all the time, it's just a, is that how you do it?
This is like that's what's on big speaker, which is our audio.
When it's just, it's going to be, if there's no sound, it's going to be great because the network's not running at all.
No.
Yeah.
Features won't change from it. There'll be no activation change to the network. It just won't update itself.
Yeah, because basically the first layer itself, nothing will change, right? And so you are not updating anything from then on. I mean, it's a little bit why people like SNs, right? There's no spikes, they don't have to compute, right? And so it's
so I'm assuming this is FPGA, right? And and this activation function you have to write to some memory, correct?
Yes.
So how do you, because it has to be on runtime. So at runtime, you have to find out the whole batch and then you have to feed it to the
take states from the off-chip memory.
Okay, so that would be very tricky, right? Because
what's the question?
No, he's talking about when you have to fetch the activation from off-chip because your network is large, right? So then
but it's a very small activation. They're vector activations. Here you have 1,000 activations. The weights are big, right? That's 1,000 square. Sure, that's a million weights.
What I'm saying, when it's an activation, even if it is one activity out of 1,000, right? So you're picking on which one and then you write to the memory, or because you have to write the whole vector, right?
Yeah.
Because then
no, you don't have to, unless you can index.
Yeah, so like there is kind of importing some kind of compression.
You need a special controller block that's very smart.
So you need some sparsity in between.
Yeah, between
this importing.
Yeah, correct. Yes.
It's all explained in great detail in that smartest paper.
Yeah. Okay, and so what we did a couple of years ago is to put everything into this new design to do keyword spotting. And so already there was a chip before that did voice activity detection, came from Min Hao, went to Columbia, and basically on this chip, he had 8 cochlear channels only, so basically he dropped everything non-binaural map, and then he put a binary MLP behind it, and the whole chip runs 1 microhertz. Like, so we're streaming constant. And then later on, of course, we did our chip. This is Quantic King and Chang Gao who did this design. And so it's got a front end, audio front end, there's 16 channels, time domain feature extractor. And then we have the RNN running behind it, and the thing here runs 23 microhertz. Yeah, it could be better, but it wasn't optimized. But it's brilliant. Now, this, I don't know if I want to do this to you guys. So you can tell.
Put up the table at least so we can look at it.
Yeah. So, when it's just when you submit a design to this kind of conference, right? You have to compare yourself against the different designs that have been published, right? At the conference or also in the journal version with the Journal of Solid States records. And so this work of building audio ASICs that do keyword spotting or voice activity detection is actually quite big in this community, right? And they're actually hitting very, very good numbers also. It's good to compare yourselves, right? To see how right you are sitting. And so it's almost like the zoology of stuff. And so I'm just going to give you this impression so we can see what people are doing in terms of finding a solution, right, to build this audio ASICs. And so for this part here, I only have this is to show you that I'm going to show you the feature extraction numbers, right? And so first of all, you have the different papers that you want to cite, then you have the affiliation, where they come from, and you can see there are certain groups that are doing a lot of work in this area. Somebody here cites from Stanford. And then you can see sometimes from the microphone, sometimes they have an on-chip ADC. Sometimes the power numbers don't include the ADC because this is a good thing to do because you count everything on the chip. Sometimes people put their chip, the ADC off chip, so they don't have to count it in. Remember, I said the power numbers on the ADC is quite good. So they actually don't include it. So you always have to read this table carefully. And so, and you report also on the process that you use to design the chip, the area per channel, what kind of topology the filter uses, number of channels, power, frequency range, frame shift, dynamic range, and so right. So Penny, I should what's the power number of microphone takes? The power of a microphone itself, a JFET, for example,
Well, it used to be really big, but these microphones with a little charged films, electric film. And then there was one of these transistors, very low noise transistors with the JFET channel. What's it called?
What's the JFET? The JFET junction FET.
Junction FET, yeah. And it used to be really big because they would burn lots of current through. They weren't worried about. I think the new microphones are much lower power.
Yeah.
Segmented,
like MEMS case.
Completely. The MEMS microphones burn a lot less power, but it's still substantial. It used to be I'm out of date, but it used to be in a hearing aid. The microphone would consume about 15% of the whole smart hearing aid power.
And this what I have.
Yeah, so it's pointless to have a 1 microwatt keyword spotter when your microphone's 20 kilowatts.
That's what I'm.
It's not so bad. It's not so bad. Yeah.
Yeah, it's true. If you tell them that, they don't like you. Because I did that in one of the forums. See, why are you serving more?
No, but nobody really knows what's the physical limit for microphone power.
Yeah.
What is the fundamental physical limit for sensing sound? I don't think there was any good theory. I looked for it a few years ago, but I didn't find it. If you have a membrane of a certain area that the sound causes it to vibrate, there's going to be some noise on that, just thermal noise, just physical noise. And then you have to sense the vibration somehow. There must be some underlying physics that puts a lower limit on the energy that's required to achieve a certain. Well, I'm saying no point.
It's an hour. Yeah, that's true. Yeah, I don't think we know enough. But we should but still, yeah, talking aside.
Yeah, is it beneficial like to go back to some kind of Giulia D'Angelo like the here usage, right? Mechanical resonate. That splits the frequency mechanically. Is it possible for them to do that? Nobody knows. And maybe it's not practical.
No, I mean, like, never best really.
So people have played around with that. You can't put it somewhere else.
Spam speech. Well, people have been doing. I've seen some people.
Yeah.
Space.
Yes, last week I got she's kind of tolerated, also not tolerated. Using your morphine. But they were trying to build a max code play and then they didn't bother to build electronic. But they couldn't get two channels.
Activate two process technology just for that. We can't even do it silicon, right?
Yeah. Yeah.
Cheaper, a lot cheaper. Takes about.
Okay, I want kind of put you guys through this, but if you're kind of a designer type, then get kind of nice numbers. But the okay, so here's to show you that the analog ones, so this is from me how you can get very low numbers, but here I just want to show that the digital ones, people are starting to come up with designs that are also going down to nanometers, right? So it doesn't mean that if you do analog that you're necessarily going to be the lowest power, right? So just keep an eye on them also because the technology is changing. Okay, and then if you want, there's a beautiful table. Metrics for voice activity detection. So one thing that actually in the community they've done is that it used to be like, early on, right, people would choose whatever data set they want and then they report the accuracy numbers and so on. And sometimes they will put like a background noise like that. And then every design that comes out, then they use some other data set or language speech, so on, and they take the noise from different data sets also. But with the keyword spotters, I think there, so I'm going to skip over that. Is that people have basically settled themselves on this data set called GSTP, Global Speech Command Data Set. Okay, it's not used anyway in people who do algorithms. And the thing that we always try to look for is that, so here's the, okay, again, you have all these numbers that come with the ASIC. But then you have to also look here because accuracy is something very important. How is very important and area is very important. So those are three things they care about. And also when people report accuracy, sometimes you also have to report the number of classes that they're trying to classify, right? So it could be that you get good accuracy for 2 classes, but if I ask you to use all 10 or 12 classes, then the numbers would drop. And in this community is, they want to see anything above 90%. So even if you recognize 12 classes and get 86, you don't like that. So just telling you, it's very strange sometimes,
And also, I keep showing there's something very big about this because the most important class of all, the real application is to not have false positives.
Yeah.
And that false positive rate is usually the dominant one. Like every other sound that's not a keyword. It's a vast universe of possible sounds, right? And this numbers, accuracy numbers, really need to see the confusion matrix.
Oh yeah, yeah.
So you'll see that in that confusion matrix, you see the unknown classes, firing false positives all the time. So it's not really this. Yeah, that's the perfect.
Yeah. So this is from our chip. So we have this. So these are the classes: silence, unknown, down, go, left, go, on, up, right, up. Yes.
What do you want to see there? See the unknown is green. It's like only 80% accurate, right?
Yeah. So here you go. Unknown. So then you could, for each of these labels or these keywords, you can see how good you are. And unknown is the lowest.
Right. So yeah, it's hard to evaluate what would be the true, but in this room, what would be the false positive rate? It's really it's sort of fake in a way because these networks that people use, the capacity of the network's not big enough to really reject false positives.
Yeah,
it has to be a much bigger network to really reject false positives. So, but everybody's kind of on a equal playing field as long as they use this one thing, but it doesn't mean that these things can be put on your refrigerator or something. But I mean, you let them on, they'd be like constantly doing stuff, It's you don't want them to do it.
Okay. Also, there is this one design that was reported in. I just point out quickly, because for Venice and we can And actually this group, again from its Middle Cambodia, and so they actually put an SNN behind it. I just show that there's. Okay, I test this, and so I give you just the outlook. So where are we going with all this stuff? Well, we're trying to look or tackle also networks that do other kinds of tasks. So here, this is an example of why we want to consider networks for different tasks, because one way of saving power is that you don't engage networks for doing speech recognition right away, right? So you want to engage ones that, for example, look for voice activity first, because then you can run just small network, also burns the least power. And then once this is true, then you look for the speaker. Yeah, and then you can burn this extra bit of power, and then you could do keyword spotting, burn this extra bit, and then finally do a simple speech recognition or send it up to the cloud, and you burn that much power. And so you can estimate how much you save by just doing this kind of hierarchical gating, we call it, right, of tasks versus just running this all the time, right? Then you can burn more. Yeah. And also this was done in a chip from Marion Verhest over at KU11. And so here on this design, they had a sound detection and also keyword spotting and speaker verification. And so what they show from the chip is that, you know, you burn this much power with 50%, right? But just doing sound detection, and then when you do the next step, then you burn a bit more. The next step, you burn a bit more. Yeah. And of course, what are we going to bring from, you know, this whole processing that happens in the auditory pathway, right, to the circuits that will double here? And so this is the other thing that we are trying to look at. Yeah, but more in kind of in the flavor or principle of what we want to do is to make sure the chips actually working in the real world, and then we actually show the benefits of the principles that biology uses, not necessarily true SNNs, but using some kind of information that will lead to this low latency, low power systems. And then just to acknowledge our networks. Okay, and then you guys. Okay, any questions? One hour. So there's one. Yeah, sure. Let me see. Yeah, it's the next stop. Okay, so guys, the title is "What's catching your eye?" I will tell you a little bit more later. I'm Giulia D'Angelo, and at the moment I'm a postdoctoral researcher at the Tech Technical University of Now, what's catching your eye? What I will talk about is visual attention. In a specific way, don't think about transformers, I don't do that. I do by inspired the neuromorphic system to model that visual attention mechanism. So nowadays, we are used to think that robots will slowly live with us, right? And then what we need is them to interact in an unconstrained world, right? So the question is, what's the first thing that a robot should do to then interact with the scene? And that, for me, was visual attention. So we need a robot to be attentive, to be able to react quickly to what's going on in the visual scene. Okay, then let me. But the big question is, how do we define attention? So why we look at specific spot of the visual field, right? It's very difficult to respond to this question. Then let's see this video if it does. Okay. Okay, the audio is not playing. I'm gonna tell you what's happening, and then we see the video, okay? This video will be played twice. You try to understand what's going on and what changes between the first video and the second one. Okay, did anyone spot anything different from the first time that I showed this? Nobody? Okay, that's it. Great. these are actually two different people. So the point of this video is because if I would have shown people playing with the balls and then the gorilla was walking by, everyone knows it. This is a bit different from all of the attention videos that I've seen. But the point here is that what was important in the scene is someone is picking up the call, okay? So this is the informative information of the visual field. What's going on, okay? And this is what we do. We try to understand overall what's going on, and we don't look at the details. Then this is also shown by Yambus et al. In 1967, he was using this nice eye tracker on real eyes of people, I can't even imagine, to track the eyes movements. Okay, so these are just eye tracking recording. And as you can see, we have different images. Okay, so it was just asking the subjects, free viewing, with a task, and so on and so forth. So imagine free viewing, we can see, for example, that here there are different people. And then what are we doing here is basically nosing. Are they discussing? Are they having a fight? Are they talking? What are they doing? What we do here is easy, triangulating. You're trying to recognize the face. What we do here is all foliage, bare bark. Okay, this is what we do here. Then again, after many experiments, Yambus got to the end saying, okay, I don't really understand what attention and how attention is driven. What I can tell you is that it tried to get the most informative information, let's say, gets the point that gives you the more information of the scene, which is not enough, but is a start. So here we have an image, again, from those experiments, and then here we have a scan path. The scan path is just a recording of the eye trajectories, okay? And then here we have a concretization of the scan path, therefore the saliency map. This means that basically what we see here is where we fixated the most. There are different models that do this. Let's take the idea that here is where we fixated the most. Therefore, these are the most salient points. I won't say interesting because it implies a cognitive bias that we don't want to talk about for now. So they're just saying it. Okay, someone, and it's not just someone, it was Loreniti, Christopher Koch, and Ernest Niebuhr, that is around, came out in 1998 with a model of visual attention. So what basically they were doing, they were extracting different features. For example, this morning, Michael Berry was talking about the bottom-up feature extraction in visual attention. This is it, okay? So you have different features that are extracted, and then you have like the scaling variances, so you have different resolutions. But then at the end, all of the conspicuity maps, they are getting to a saliency map that is giving you this, okay? So basically different features, different channels of the information are competing to tell you what's salient in the scene. Cool, amazing, but feature extraction is not enough. Why is not enough? Because then from there, how do we really perceive the items around us? I don't know if it's one of your questions, Mike. Then everything starts from 1950s, Gestalt principles. So Gestaltism is a word that is a German word. So if some German can help me, it means whole, perceiving the entities in their whole, in their not unicity. Yeah, I need German seeing this. So basically what they're saying here is that they want to give you different rules with which we perceptually understand the scene. Okay, let's go for them. So if I ask you what's here, I'm sure you will say, oh, this is a dog, this is a square, this is a triangle. There's no doubt, okay? But why? Because the brain is eager to fill these gaps and see if there is something that it can recognize, okay? Because we are kind of comfortable with labeling things. Then there is the same similarity. So if I ask you to group things, most likely you would group circles and triangles. Then we have the proximity, same thing, you would group for proximity and figure ground segregation. Therefore, you have the distinction between the foreground and the background. Okay, so these are perceptual rules to understand what's going on. Okay, then let's stay with the closure. And as you can see, we are in 2000. What happened in 2000 is that actually someone, and this someone is Zhu et al., they found some cells, let's call it some cells, in V2. These cells, as you can see, these are the cells, and they're placed on this edge, and the foreground is on the left and on the right, okay? You can see that the foreground and the background has different colors, just to show you that it doesn't matter, okay? It's color invariant. So you can see that the cell is responding only when the foreground is on the left, because here it's just spontaneous activity. Therefore, let's take a closer look of this. If we then put the same thing that we were looking at before, then we have the same square. And then if we imagine to model these cells as a half moon shape, banana shape, whatever shape you want to call it, and then you use this shape of different orientation, then you can group the information, you can pool, so you can have a layer of border ownership cells and a layer of grouping, and you can perceive that there might be some object, okay? It is called the proto-object because we are not sure that is an object. It's an object to be. So we think that in that area is salient because there is something that might be an object. These are called border ownership cells, not for a case, because they're telling you what's the ownership of that border, okay? It owns to that. Yeah, and the priority of background versus foreground doesn't matter here. Doesn't matter that, and it doesn't matter even that scale. Yep. So this is our banana shape, half moon shape, whatever filter, which is called the Bondi's filter. Again, someone, which is again not just someone, Russell et al. And et al. Is Ralph and Cummings. It will be around, so if you have other questions, I can respond, but it's better if you ask him. He came out with this. And then we have again intensity, color opponency, and orientation, okay? But what's the difference? The difference is here, okay? That what they have done is that they added a border ownership layer and a grouping layer. As I was saying before, here you have the different orientation of the partnership, and then you group the information in the layer here, okay? So, this is a start of my PhD. Giulia, you have ICAP. Hence, you need to make him interacting with the board. And ICAP, in my case, at the Institute, Italian Institute of Technology, I don't know if you know this guy. How many of you know this guy? Okay, yeah, okay, so quite a few. So it's the humanoid robot that we have in Italy, at the Italian Institute of Technology. And in the Chiara Bartolozzi's lab, we have mounted two 80s cameras in the eyes. Okay, so we have the bio-inspired cameras. Then I was thinking, okay, which kind of information do I want to make the robot be able to interact with us or with the scene? Therefore, I wanted for sure intensity, so the raw information coming from the cameras, and also I wanted depth. Why that? Because I want the robot to be able to say, okay, this is an object and this is close to me, then I can grab it and I can do something else. Then motion. Why motion? Motion is super interesting and really important because you can do obstacle avoidance, you can catch things. So it's the third channel that is really important for interacting with the scene. Okay, this is my PhD. Basically, what I've done is that I implemented the intensity channel, the depth channel, and the motion channel. So there's one thing to be said here. So this is GPO. So I'm doing given frames. Was I satisfied? Not at all. Then I've done it for Spinnaker. Okay, so this is the same channel, just different platforms. So this is fully neuromorphic. And here, as you can see, I don't have a kernel that is convolving through the visual field, but rather I have layers of neurons that are resembling my von Mises filters, okay? Then we have the DAT. The DAT has a disparity structure before. That is basically the disparity structure is the stereo matching cooperative neural network from Conrad and Fiuzzi.
And they worked together and they took inspiration from all Oswald. There's lots of names, but if you're interested, I can give you all of the references. So we were getting this parenchyma, and then it was going into the proto-object model. And then I had the saliency model. The motion, as you can see, it doesn't go to the proto-object model. There is a reason. Because when something is coming towards you, so if I just get a bottle and I throw it towards you, you don't really care to understand that it's a bottle, you just want to run. So it's really not important that we detect or we recognize the object at the first bottom-up level, okay? So the motion is going on its own, and it's a spiking elementary motion detector. If you're interested, again, we can talk about it, but it's basically you have two receptive fields next to each other. If the first one is triggered before the second one, therefore you have a left to right motion. Okay, it's very easy. And that's it. And this is the model that I've done in the end. Okay, we have selective phenomena and intensive phenomena. What does it mean? It means that, for example, if there is an explosion, this is an intensive phenomenon, and then you react, you are attentive, but because of alertness. Instead, the selective phenomena is rather covert and avert attention, where you focus your attention for some reason. We don't know which is the reason, bottom up, top down, we're not sure, but you move your attention somehow. Okay, let's look at the depth. I just want to show you what I've done. So here in reality, so, okay. So here you have the RGB video of what's going on. Here we have the rectified stereo events of ICA looking at the film. Here we have the disparity map. Here we have, so what is important is that you look at here and here. This is the saliency map coming from the intensity channel, and this is the saliency map coming from the depth channel, okay? And let me play it. Okay, so if I have one object, of course I have the attention, which is the red dot, is the maximum salient point in the scene. Then I put one object, and as you can see, the tension here is pinpoint between one object and another one. And here it doesn't, okay? It doesn't, why? Because basically, so it's very easy. I'm basically feeding the model with the disparity map. Therefore, the higher disparity map, the closer the object to the robot, and you will get the more salience, okay? So it will be, for sure, the saliency, the more salient point, the object that is the closest to the robot. And this is kind of giving task to the robot. Be attentive to the closest object to you, okay? But this is just us. Okay, so I've done this different experiments, but what I want to let you understand is that I don't have a real accuracy. Can't have a real accuracy. Why? Here you have the input, so that are RGB images. Then you have the depth map, whatever. Then we have the RGB saliency map, because I'm taking inspiration from the model that Ralph Etienne proposed, so that is done in RGB. Then we have the event-driven saliency map, and these are real 3D fixation maps from humans. Why I tell you that this, although I have a ground truth, why I tell you that this cannot be my accuracy? Because humans are much more complex. It really is in the response. So what I do is bottom-up attention. I'm extracting features. What we do is much more complex than that. And I will never be sure of your cognitive bias behind what you are looking at. Because you might look at red, because the day before you were very interested to red, you were painting red, and therefore this will result in your eye or in your saccades, right? And this cannot be catched easily. So what I do is usually a comparison between my model and the RGB one that I use as a baseline, and then the comparison with, again, the fixation map. I try to do this comparison anyway, because it's still a validation, right, of how far I am from these fixation maps. And these are basically the metrics that we use. So I'm using all of the MIT metrics to detect how far I am with my saliency map to the fixation maps. As you can see, and as I was saying, the results are not great. So we go from 0 to at maximum 0.5, and we should, like, the best should be 1, which is the most important message I wanna give it to you. That is really difficult for me to understand the accuracy of what I'm doing. and it's a struggle. Here is the same thing I'm doing. In this case, I'm doing the structural similarity, trying to understand again if something changed. I tried to change also the overlap of the Bob-Mises filter on Spinnaker. Nothing really changed. So my results are a little bit better with the structural similarity. Indeed, the structural similarity works really well for me in general. But again, these are not great results for everybody. They're great results for me. So again, here we are at the bottom of another question, right? Humans are not driven only by bottom-up mechanisms. We said this. And how do you really distinguish between bottom-up and top-down mechanism and a human being? It's difficult because they're likely to be very interconnected. And it's very difficult to understand exactly, unless I put you sit somewhere and I give you a focus and I give you a task that you are performing. It's very difficult to understand if that is top-down or bottom-up. Okay. Then, at the end, I am with different approaches and different channels of the attention model. These are just the classical patterns that we use, the proto-object and no proto-object patterns. This is the intensity channel, this is the depth channel, and this is the intensity channel with Spinnaker. You can see these were done in the running in GPU, and you can see that, of course, with Spinnaker, we get to a nice latency, but with 6 boards. And 6 boards like this, which is unfeasible. Imagine ICAP working with the 6 big boards next to it. It's just feasible. But we hope for something better, guys. We always hope. Okay. Then I got here, and then there is my TI, Chiara Bertolozzi, that kept saying, yes, fine, we did nice things, but there's no saliency in the brain. There's no proof that we have a saliency map somewhere in the cortex. And this stuck with me. And then I started asking myself, is the active interaction with the word the response? Might it be? I know you agree. Okay, so let's ask ourselves this question. This amazing crocodile and this amazing giraffe, they're both walking, but the big question is, and we had this conversation the other day, does the brain work in the same way? Although they're walking, they're still walking, but the point is, if you just take the brain of a giraffe and you put it in the brain of a crocodile, is that going to work? Probably no, because they have a different body. Therefore, embodiment refers to the concept of experiencing the word through a physical body or form. Is your body that is giving you the perception of the word. Therefore, you understand the word for that reason. So it's really attached to the concept of the body. And again, this is a quote for my professor in Prague. You can't prove the brain of a giraffe into a crocodile. Okay. Then we ask ourselves this question, and there are 2 people that actually has done it way before me, which is Ballard, as yesterday was mentioning, Toby was mentioning them, and Camilo Regan. So I just wanted to introduce you to the sensory motor contingency theory. I don't know if any of you is familiar with the sensory motor contingency theory. Yes, please. So basically, the idea that these 2 guys have is that active vision and vision is much more exploratory than what we think, okay? I tell you why. So what we think nowadays is, okay, I see something and I have a mini representation of that thing inside, okay? That's not the case. We don't do frames, right? So we keep thinking that what RGB cameras are giving to us is like a mini representation of what's going on in our brain, but we know that is not like this. There's not a real representation or internal representation, but rather we probe the word. So we saccade. Without doing saccade, we will not be able to understand any of the things that we look at, okay? So we have to probe the word to see what's in front of us. And this is the sensory motor view. Then perception is not an isolated processing step, but searches for relationship between actions, the saccade, and resulting changes in the stimuli.
How does this relate to actually sensing?
Because you need to interact with the scene, right? With your skates. Why does not relate to active sensing?
I ask you, is it the same? Like this, what you say now, right? Is that you're not just passively sensing, you're sensing by probing the world. Is that the same as active?
It's the same as the same way they will advocate. So we need to separate because we have this here at full PR, right? The 2 degrees are sharp, everything else is not. If you have a camera, the image is perfectly sharp and all its million pixels. By doing it such that.
So, okay, yes. First of all, because I like to understand what's going on for us. But second of all, because of the data, right? Like, for example, now we have this amazing Sony privacy camera, but you need to have a certain kind of bandwidth, right? To be able to use it. And most of the time, what you cannot try to do is then downsampling. And then why don't just downsample? That's the retina does. Then the retina does it in a complex way because it goes into the log polar space, and that is math. I agree with you. But it would be cool to somehow have, and I've done it for the motion channel, for example, to reduce the resolution where it's not needed, which is actually the periphery. So the point is that if you need to kind of shape your information smaller, so you need to have a ROI somewhere that is very small. Therefore, to get the global point of view, you need to move. So the point is, in my opinion, this one.
Just one quick thing regarding active sensing. In a lot of the engineering fields, it directs us to sending energy into the scene. Say light up against the active sensing then. So showing my position or sonar and so on. So in the nice paper that was headed by Maurice a few years ago, we make the distinction between active sensing, the engineering thing, and closed loop sensing. By moving my own system through this.
Yeah, and there is also a nice again paper from Horitz where it's like we should do much more close to
Wait, but then that means then you're kind of separating sensory motor agencies. Like,
yes, and you could just have both, right? You could have the LiDAR where you're also moving your receptor to somehow do saccadal LiDARs, right? But just to separate the two somewhat orthogonal concepts, and your more identical active sensing is close to chain. Which trade constitution the rest of the insurance was.
Yeah, I'm not convinced. I mean, if you answering Giulia's question, if the problem is recognition, I don't see why we should move. But even the early days,
it's so nice because the presentation is about.
One of the papers that I can often get is introduce. If you move in certain ways, you introduce constraints to make your problem easier. For example, if you move with an old drug selection, you make estimation easier or shape estimation. That has been demonstrated, and that is also seen in animals, like, some rats, also they jump up and down, they could use slow motion. But not much of this yet, right? I think in the field, like purposeful moving things. But using lowering, for example, if you say, just talking computationally, we know that translation and rotation becomes, for example, that detail each other, which makes the 5-dimensional work.
I'd like to read more.
Oh, that was 7 years ago, and we actually have many papers.
Thank you.
Because it's even with the best camera, the senior guy consulting experience, they can actually do this. You know, I say they cannot see the back.
Yeah, it's actually we will see it at the end of the presentation, but that's also the point. Okay, so we got to this point of probing the word, and the point is also the fact that basically, if we look at a painting, right, and our attention is focused on the chin of the lady, and then we moved our attention, and then we changed something on the painting. If we had this internal representation, we should be able to somehow subtract the two things, but actually it does not happen, okay? Because this for us is a proof, and us, I mean, who thinks together with Ballard and O'Regan, that actually the outside word acts as an external memory, therefore is a store for us, where the information is available for probing by means of eye movements or attention. So we don't need to represent internally the things that are already here. We just need to move our eyes. Okay, so we have attention. And then we have two guys, Mayer and Angel, that try to understand how to perceive an object and classify an object because of the movements and the relationship between the movements, the actions that the robot is performing, and the visual input that was changing. So basically what they did is that they had the sensory observation, so the visual input, and then the action that was basically them walking over the objects. And then thanks to this relationship between the sensory observation and the action, they were able to classify objects. So I took inspiration from this, and I started from this, which is actually the idea of the paper and the idea that comes from everything I told you so far. So basically what happens is that the sequence of actions and sensory observation should give you the information of what we are looking at. Therefore, our circuits and the movements that perform in the scene should be able to be enough to classify the object. Yes?
It's on my desktop here. I don't know what to do with it though. I read the article you would say. That's it.
My bad. Well, switching off.
Are you acting to the keyboard?
Which here's that?
Oh, I mean, it's muted again.
Okay.
You got it.
Okay, therefore, this is my idea. And I will not show you results here because I just started the project. And this is the idea behind the project. So what I'd like to do is trying to take information from how the infants are actually learning new objects through saccades and wrist movements. That is connect to what Michael was saying before. So we move our wrists and then we move our eyes to gain different points of view, and therefore we learn what we have in front of us. So I'm taking inspiration from infants with, I mean, of course, working on ICAP, and then I have other problems. I have other problems, for example, in motion suppression, because when we move our eyes, we know that with the cameras, when you move the cameras, then you have this bunch of events coming towards us. And how we do it in our retina, or anyway, from the retina of salamanders and rabbits, is that you suppress that motion, you suppress that firing thanks to the amacrine cells. And the amacrine cells basically are spread all over the visual field. And what they do is they work in time precision. So if all of the visual field is triggered at the same time, this means that probably it's you that you're moving. And then I'm taking inspiration from this, trying to understand if this could be helpful when I am saccading. And this is actually the pipeline that I built, and I try to understand. I'm trying to understand how to really tackle all of the problems. So it would be also nice if you have any ideas, or if you think that something should be done better, please come to me and talk to me, because I really need it. And this is what I will do. So I will take pan and tilt movements of ICAP, and therefore I will take also the wrist movements of ICAP, and this will be my actions in the pipeline. And then I will move, thanks to attention, over the objects, and I like to classify the object. Then I have this.
So my talk happens to interlace with Giulia's quite nicely, and in terms of saliency and kind of mapping of things. Yes, I'm very pejorative in my title. That's not true. I'll maybe try and explain something better, but I'm Paul Kirkland. I'm from the University of Strathclyde for the next 3 weeks, and then I'll be in Western Sydney University. But I'm just going to try to explain a lot of the work that I've done from a different perspective from Giulia. So we actually started roughly about the same time, but didn't know this, and have two kind of weirdly similar paths of trying to solve the same problem. But I come from an engineering background and very algorithmically tackled the problem. And I just stuck with STDP because by time-dependent plasticity was fun at the time, and there wasn't as much research on it. People had seen the limitations of it, but I wanted to see how far you could push it and see if there was a useful application for it. Yeah, take the forward. So the first thing I tried to do was UAV tracking. So this was a popular thing to try and do at the time, because UAVs were becoming mass popular, and how can you actually track them through space and time, because they have really relatively small profiles.
but they're highly dynamic, so you can't just have like a ballistic tracking algorithm on it and go, oh, well, I know it's going to go next. They're very, very dynamic in their movement. So could you pair an event camera with a neural network that could work fast enough that you could actually track them in real time, that you could actually follow them around very accurately? There's a lot of latency in the so what I tried to do was build a spiking neural network using spike time dependent plasticity. What you're going to notice is that the features aren't very spatially representative. Welcome to spike time dependent plasticity. But you were able to identify enough features that you can identify rough object shapes of like the DJI Phantoms and other ones that we would use during the trials. And what we were able to do is actually be able to pick out those objects for a relatively small spiking neural network, but actually propagate all the information through relatively quickly. Also, these networks are only trained to have weights of 1s and 0s, so we're trying to make everything quite sparse and minimalistic when we're actually doing all the processing. And then what that leads to is these features can then be mapped back onto the drones as well. Please, come on. I can see it on my screen, this is the problem.
Those features were just learned with the STDP and you're just plotting like the weight.
Yes, so this is a saliency mapping then. So similar to what Julia was saying, this is just saliency mapping of me seeing what features were active and when, and how they actually relate to certain parts of the objects. So in drone tracking, it might be useful to track the propellers or the battery because they're quite obvious parts to look at if they're wanting to maybe deal with drones in certain ways. But then what you're able to do is actually able to do this saliency mapping on top from the event camera data. And doing this saliency mapping then led me to think, well, I can do this saliency mapping backwards through the network. I could basically just map this back to the pixel domain, and then I could actually probably just do segmentation. So then I came up with the horrible idea of doing STDP trained spiking segmentation network, where half of the network, the encoder, is trained, and the decoder is just taking the spiking activity map or delay lines, depending on how you want to do it discretely or continuously, and then mapping that back into the pixel domain again. So that information comes in, and if faces happen to be a class in your network, then that's the things that were most prominent or most salient for the face features. So why would you want to do this? It might just be quite useful to do tracking at a pixel level. So that's why I tried to propagate forward to do tracking. And you could combine this into bigger systems to do control or light control in the systems where you actually have perception, understanding, and action. I don't want to call it cognition because it doesn't feel like cognition. Why is my delay so bad? Oh man.
Here, is everything still learned by STDP?
Yes, only this part is done by STDP. I swear, honestly, my screen is on the next screen. So.
Can I ask a question?
Yes.
So what did you use as your training data for these networks?
So for the drones that we collected, just our own drone data. For these ones, this is now the Caltech data set. So the Caltech data set that got captured by a neuromorphic camera doing the little triangular saccades. But you're going to soon find out that doing spatial representations with STDP is actually quite challenging and is probably not where you should use spike time dependent plasticity to do like spatial feature extraction.
Was it all like pre-labeled, like your drone data?
No, this is unsupervised, so you can just do this unsupervised. You just throw information at it and then see what it clusters and sticks together by seeing information together. So as I was saying, you can put this into a control system that could be quite minimal then, so you could actually propagate information through the system and actually kind of do something with action, so you could quickly move things around in a closed loop system. We also realised that the more information, well, the more diversity you can put into your neurons. So if you have leaky integrating fire neurons, so a lot of this work started from a derivative of Simon Thorpe, Timothy Mascaleer's, and then Sai Kaderkis's work. So it's building on top of all that idea of how you find little proto-objects in the scene and try and build it up. But they only used integrating fire neurons. But if you use heterogeneous, leaky integrating fire neurons, you can get much better spatial representations. Still wouldn't call them perfect. But they're really interesting in the sense that you can actually map, because you're using spike time dependent plasticity and you have an event camera, you can map the saliency directly from the network that you have into how it came into your neurons. So you know that saliency will typically mean that the spikes fire earlier rather than later. So if you have a moustache, like lots of the people in the Caltech dataset did, that might be a salient feature to then help identify things. And just to visualize that slightly easier, this is what actually happens when you're propagating your information through the network. You can see videos are not synced. You can see it propagates through the network into some sort of classification layer. So going through convolutions and pooling, and then can go back through into the output again, just highlighting the features that are the most salient for faces.
What's the color?
Colours are just the features embedded, and so from the previous ones, all the colours were just represented in different features. So these always represent different features, and they get colored.
Sorry, am I right in thinking that it's completely reconstructing the output face from the convolution 3 class layer?
Yes. So everything that caused these spikes to fire via the pooling layers and were like the first spikes to fire is then what gets propagated backwards. So it's not really segmentation in the true sense of learned segmentation, it's genuinely saliency mapping. But saliency mapping and segmentation seem very similar in this context and overlap an awful lot.
But you also had a skip connection, right?
Yes, the skip or delay connection is to ensure that there's no uncausality in these. So if I actually just propagated the convolutions that existed in this space, it would be more complete representation than what's there. So you'd have to know if you have a 5 by 5 and you have like 10 kernels of that pixels that would have to be active. You want to make sure it's those 10 ones or 9 or 8 or however many it was exactly. So you're not trying to hallucinate or imagine that there's things that you didn't see in the event space. You're only causally allowing the spikes that did fire to fire. So you do that with delay connections or with a spiking activity map, but that's
But then coming back to Sebastian's question, right? Then I mean, do you have an intuition for because if you're reproducing, like the output here is very close to the input, right?
Yes.
So do you have it? Maybe I misunderstood what you said before, but do you have like an understanding of what part of it is actually the classes and what part is the skip connection? Because you could, in principle, just learn.
No, the skip connection can't do anything. The skip connection just inhibits or doesn't inhibit. So the skip connection sole purpose in life is to just inhibit the connections that aren't useful, not actually propagate information through the network. And implicitly.
But you're still inhibiting, right? We're just technically propagating information.
Well, yes, there's a spiking activity map that's like an inhibition map essentially that gets passed through the network. And if that falls into your inhibition map, then it can propagate information out.
Okay, right, yeah.
So there's no active information, it would just be the information you couldn't use, yes. Oh, my internet connection is clearly not, this is the issue.
Hey, Paul. So I got a question here. So on the slide that you showed right now, you basically are not training in a supervised manner, right? You said, and the follow-up question to that is, you have convolutional layers and they probably have parameters.
Yes.
What are you just initializing it intelligently then? What is the secret sauce?
Yes, heuristics are the secret sauce. So there's an awful lot of thinking about what you're doing and figuring out what the receptive field sizes are and stuff like that to kind of make this work. Like that's the big problem with spike time dependent plasticity is there's a lot of handcrafting before you do anything, which isn't useful. It might be useful in some senses because computer vision existed for a long time before this came around, and there might be useful things that you could actually just physically embed into the network rather than it having to be a learned process.
I see. And last question is for the decoder side. Is it just basically an increase in size of spiking neurons, and the last layer is just a bunch of neurons that emit spikes? What's the decoder structure? The encoder structure I can imagine, but the decoder structure.
The decoder structure is just the inverse of it's just the inverse network, and it's just the activations are still the information getting propagated through. But there's nothing, the only thing that's stopping it from propagating is the spiking activity map. So the information that was contained on what spikes did exist in the encoder would allow only those spikes to exist in the decoder. So it's just like a noise filtering or filtering process essentially is what you end up with at the end.
Got it. Thank you.
It's just semantic filtering, which is a weird way to call it.
I see.
To the last slide, I just have one more question. So I didn't completely understand. So does the collection of pixels in the class 3 class section, that represents the class that the neural networks extracted from the output, right?
Yes.
So if I have a different face in the input,
yes,
will it show the same class? Because
Yes.
it's also a face.
Yes, great question. Let's explain why that's useful, because this is what it's going to now do. So that's semantic segmentation. But if you want instance segmentation, you would have to then be able to identify things within the same class. So the way I tried to do that within the same class was that spiking activity. So a raster plot of when did the spikes occur and which features occurred in which order. So you can actually map them into a feature time map, which just becomes a raster plot essentially. And if you look at that raster plot, you can then actually, this is backwards. You can look at that information and extract a certain person's face out of it. And you can even do that within noisy data or doing like difficult tasks like occlusion, where people disappear behind each other. So you're actually able to do instance segmentation just from doing the saliency mapping. So what was the spiking feature order essentially?
What's the difference between instance segmentation and semantic segmentation?
So semantic is like class building, so face would be that representation. That instance would be within a class, can you identify people? So it's a big computer vision one of like, if you can identify cars, can you identify individual cars? Okay. Or dogs, can you identify an individual dog and a scene of dogs? But yes, so what I'm trying to get to was that if you do instance segmentation, you're able to extract all the people's faces, and if you find the closest match to that person's face from your spiking activity map, or your raster plot map of your features over time, you can then map that to the closest person, and you find the closest person. Hurry up, Mr. Slides. So the next slide is trying to show you this this map as a representation of it, mapping it all the way through. But this is kind of where VSA comes into some of this now, because there might be a useful way to take the raster plot of your spiking activity and convert it into a vector symbolic architecture, or could you use...
Are you on a VPN maybe? Is it possible that that's slowing you down if you're going through Strathclyde?
No, I wasn't on a VPN.
Okay.
I share a presentation with me and I share for mine. That's a
Paul?
Uh-huh.
Hi, Paul. A voice from the off. Maybe using the gap here for a short question. So you said that spatial feature extraction with STDP is maybe not the best solution. So could you imagine alternative approaches?
Yes. So some of the usefulness of spike time dependent plasticity was the features were very understandable. Whereas if you use deep learning, sometimes the weight maps that you learn are slightly more complex and obscure because they have multiple realizations and multiple abstractions in them. So that was the main part of why we tried to use this, because a lot of what I tried to do was interpretability, so that you could actually understand why the network classified something or featured by something. I know you can do saliency mapping the other way around with deep learning, but a lot of it was just to try and push what spike time dependent plasticity could do, because there's meant to be lots of hardware benefits of if you had a really sparse in-time network. When you try to map that to hardware, you might get more sparsity or power savings or latency savings, essentially. And I have no idea.
Okay, yeah, thanks.
But yes, to get to the more interesting parts of the talk of where we're going next. So I don't think it's very good at doing complex spatial reconstructions or understanding, but very sparse spatial temporal identifications do seem like a much more useful field. So if you're trying to extract all these pieces of information and trying to follow all these dots around, it's a relatively complex task because all the spatial features of all the dots were identical. There's no differences between them. So you must have to track their paths. And because they go over each other's paths, then that can be slightly more difficult for Kalman filters and other filters to try and actually map and follow them around, unless you know something about the embedded motion. So over time, that would just look like a creepy creature from Stranger Things. And if you look at that information through time, it's much easier to be able to extract everything. And if you could learn the representation, so this is very bad contrast, but it's just showing the network doing an event-based and an accumulation-based build-up of what information gets propagated through the network, so that you could actually reconstruct what the 6 motion paths actually were through learning it over time. Yes?
Can I ask, how did you create this data set with the 6 dots moving around?
By recording each 6 individually and then overlapping on top of each other in the event camera data space.
Were they drones or?
No, that's a laser pointer moving around on a magic gimbal that costs far too much money. So to spiral a laser pointer around. But so it was very repeatable. But essentially what you can do is reconstruct where all the motion paths led to, and then you could follow everything individually from where they were. This was just to try and be able to recognize different features through spatial temporal information or spatial temporal feature information, as in what features existed and when, and to which of the track paths that it belonged to. So the usefulness of this is, the bandwidth is terrible, as you're trying to track that point through space. If you try and do that with a Kalman filter and this incredibly noisy scene, which is like -10 decibels, Kalman filters and particle filters really struggle to do this task. But hopefully you'll be able to see with the SNN, there's just does a great job of doing like noise filter removal essentially. So that kind of led me to the point of, well, I kind of thought the segmentation networks through saliency was just doing a good job of filtering. So maybe it does a good job of filtering things that move compared to things that don't move, or so in this case just noise. So background shot noise versus actual movement in the scene.
Can I just double check something? So that's again with your encoder decoder.
Yes. Everything's always encoder decoder STDP. That's why it's all you need.
I didn't get how you managed to differentiate between the different trajectories or the different dots.
So, well, I was just trying to quickly go over it. There's too many slides for that. It's looking at little features as in space-time features, as in did they move in a figure of 8 kind of motion, or a ballistic path with a strange ending to it. So it learns those little representations of features. And it depends on which one of those was most active. And then combined them and goes, oh, wait a minute, it was this motion path I've seen because it looks most like the figure of 8 at the end.
But you're doing this at the end of the recording?
Yes, so it's not like active tracking then, which is the sad point of it. This is where I'm trying to get to with some of this. This might be a useful case for doing space situational awareness then. If you take Western Sydney's data, which was the EBSSA data, you can do a similar task to try and do their satellite tracking, but you notice in the satellite tracking task that there's a slow moving object and then a fast moving object in the scene. So you want to be able to identify both of them simultaneously, and be able to track all the other objects that will randomly fly through the scene quickly or slowly. So to do that, you want to split up fast motion and slower motion. So you need a wide range of leakage factors in your network to be kind of receptive towards things that might be a lot slower and not moving, or things that are very fast. So then you'll create a lot of events over a short period of time. And similarly, you can do the same thing when you're trying to do space situational awareness. You're looking for things that are going through the scene, either slow or fast, and then you can classify both of them. So the yellow thing is satellites that were moving fast through the scene, and the blue thing is a stationary object in the scene. And you're using the same type of network setup, same STDP. The convolutional things make no sense now because there's time elements in these, which you can't see and I didn't visualize very well. Maybe I could have went from light to dark. But that's what you're looking at, is it's picking up all the different things from the scene using spiked independent plasticity learning. I'll skip this video. So what I've tried to move to more recently, and what we're trying to do for my last project essentially, and maybe projects I'll start at Western Sydney, is enter useful ways. Obviously there's lots of people who are working on optical flow just now, but a lot of that optical flow isn't sparsely related to the scene. So is there ways that I could do this from the spiking network itself in each convolutional layer? Could that actually start to represent heading, direction and speed? And could I do that on multiple layers of it? So then the featural representations could also have that information encoded. So you're not just doing it from pixel basis, you could do it from featural basis as well. So you can look for corners similarly, or more complex things than corners, like actual meta parts of the objects. So how can I actually map that into the network at the same time to propagate all that information through and do a little bit of either optical flow analysis, but try to get that into a tracking perspective so that I can actually identify the object from its movement paths, or the kind of vector of movement based on top of the information that's there. So this was just a quick, simple example of doing this, but if you try this with Lucas Canary or other optical flow methods, it sometimes gets a bit distracted by the backgrounds, or just the fact that event cameras have a weird tail to them all the time. So the objects always have long tails. It gets a little bit confused in that, but I try to be as nice as possible. The Kalman filters, particle filters, and the optical flow methods, I put like minimal effort into. There's obviously optimizations you could do to this. These are all just kind of basic implementations from MATLAB, but also I don't try and overly optimize the spiking neural network, and it just works better in some cases.
You just mentioned those other classical algorithms used with things have long tails.
Yes.
Is that because you're processing in a frame-based way? Yeah, right.
Yes, because how do you feed that information into some of these algorithms is not like it's not an easy way to do it. So Kalman filters have this problem where I try to blow up my computer by trying to feed it into a Kalman filter event by event, and then you realise it just tries to explode because the sheer computational power it tries to do that on an event basis isn't efficient. Obviously, there might be efficient ways to do it, but if this is an approximation to some of these, then this might just be an efficient way to do it in spiking neural networks. So, but the bigger opening question is, algorithms need to be developed for event cameras for doing the similar tasks to what we already have traditionally. And this was just my attempt to do it with spiking convolutional neural networks, because currently these are quite optimal to use on a GPU basis. So I could do lots of these at the same time, rather than trying to do like looking at a small part of the scene and doing any of the processing on it. I could just do it on the full image all the time. And as long as none of these are processing anything useful, there'll be no information being propagated through. So hopefully it's computationally relatively sparse. So
some of the most challenging satellites that we have broken down tracks where you get few events, then they stop, they reappear again, and so on. And that's typically challenging for tracking, conventional tracking algorithms. You end up with multiple tracks and to merge them back. It's tricky. Very quickly for local descriptors as well, because they see only two events and that gives like noise, and it stops looking like noise when you count all of the thing. Which we solve with something like, say, contrast maximization. It's not terribly elegant, but it's very brain-based and takes the full data. Is there a natural way to do that, perhaps with multi-scale SNNs, or do you think that that's going to be challenging as well?
No, so from doing this, with some of the EBSSA data, there were some ones where the track paths weren't very nice, like they weren't fluent the whole time, and it was able to deal with them just as well, as long as you assume that the highest, like the widest field of view from your convolutional neural network combines all these little paths, like the smaller 5x5 paths or 3x3 paths. That's what it looks at initially, is either 5x5 or 3x3 space. It says, was there enough like signal in this to cause an event? Then hierarchically, it's how many of these can I stick together to build up that shape? So you can build up shapes that are like oscillatory, so they come on and off. You can build that up within a representation of it. It's a learned representation that would have to be learned, or you could force it to learn that.
So what if there's no shape at all on the first layer? Basically, you have single events completely isolated, even in a 5x5 window, and you start seeing alignments when you go beyond that.
Yes, so that's the one thing I can't do currently. You'd have to do that in a weird like reverse scale, have to look bigger and do it first. But typically for most of these ones, that didn't seem to be as much of an issue. This is why I'm coming and doing some study to figure this out, to see how much of that is true. Because in most of these examples, the signal and noise is quite easy to differentiate as long as you look at the smallest time frame you've got, because it does occur as like little chunks compared to the noise. So
realistically, like 95% of the satellites do fall in this category. The question is,
5%.
How do we optimize for the remaining bits? And that's where it becomes tricky.
No. So yes, that is all my slides anyway, so I can be on time roughly. So thank you very much.